Okay, you can start now.
सुरू करू का सर?
Yes.
भावेश जॉईन नाही ना झाला अजून.
भावेश जॉईन झालेला आहे, स्टार्ट करा.
हा ठीक आहे. Good morning, everyone. Myself, Mohite Sakshi. A warm welcome to all of you on day 2 of third annual Swalife International Research Conference on Innovation in Drug Discovery. After the inspiring session we had yesterday, we are excited to begin today with a fresh energy, enthusiasm, and more enlightening discussion ahead. The first day of third annual Swalife International Conference and on innovation in drug discovery was successfully conducted under the able anchor of Sanjana and Shreyash. The event was organized by Swalife Foundation in association with Swalife Society for Translation Science and Innovation and Swalife Biotech Limited. The day started with a warm welcome to a global participants, followed by insightful keynotes presentations by distinguished speakers. Dr. Praveen Bade, founder Swalife Biotech Limited, Ireland and India, who spoke on personalized herbal formulation in chemoprevention of oral cancer. Second one, Dr. Irigo Di Otto, founder of OACP Limited, Ireland and Italy, who presented on genetic biomarkers and fish accelerating drug discovery innovation. An engaging poster presentation session followed where researchers, academicians, and students presented innovative studies focusing on cutting edges topics such as AI in drug discovery, natural product innovations, molecular docking, network pharmacology, and nanotechnology-based drug delivery system. The first day concluded with active participant from attendees and positive feedback on enriching knowledge exchange, fostering collaboration between researcher, industry, expertise, and scholars. To begin today's academic journey, we have organized a highly anticipated workshop on network pharmacology. This workshop is specially designed to provide our participants with a practical understanding and hands-on experience of the latest research methodologies in the field of drug discovery. Workshops like this are crucial because they bridges the gap between theoretical knowledge and real-world application, equipping us with essential skills for research, innovation, and for future career growth. Over to you,
That's
Bhavesh.
absolutely right, Sakshi. Today's session will highlight emerging computational technique in pharmaceutical science, including protein ligand docking and PPI mapping, cake pathways, ADME screening, and much more. It's going to be highly interactive and collaborative, giving participants the opportunity to ask questions, share ideas, and engage directly with experts. So, dear participants, we encourage you to stay curious, participate actively, and take full advantage of this valuable learning experience.
We would also like to express our sincere gratitude to our organizing partners, Swalife Foundation, Swalife
Swalife Foundation, Swalife Society for Translational Science and Innovations, and Swalife Biotech Limited for creating such a impactful platform where students, researchers, and academicians are industry experts come together to interactive, share knowledge, and grow. Before we proceed, a gentle reminder to all participants, please ensure that your mobile phones are switched to silent mode to maintain a focused learning environment. Also kindly keep your notepad ready because the insightful share by expert facilitators today we will be extremely valuable for your academic and professional goal. So without further delay, let's us officially begin the workshop on network pharmacology. We hope it will be an enlightening, interactive, and an enlightening experience for everyone present. Stay tuned for a power pack session filled with expert knowledge sharing and practical demonstration. Allow to introduce Mr. Harshad Pisal. He is a postgraduate researcher in pharmacology, currently working as a research associate at Swalife Biotech and as a trainee intern at IPIS Intellectual Property Services. Mr. Harshad is actively engaged in vitro and in vivo pharmacological evolution, network pharmacology, molecular docking, formulation and development, patent drafting, and regulatory documentation. He is published and present research in areas such as herbal actives, nanotechnology-based formulation, and network pharmacology-driven drug discovery. His core expertise in bioinformatics, preclinical researchers, and scientific writing and data analysis. Without further delay, over to you, Mr. Harshad Pisar.
Thank you, Bhavish. Thank you. I would like to thank Swalife for giving this opportunity to deliver the workshop on network pharmacology and molecular docking. So I'll be delivering the workshop on network pharmacology, where I'll show the live demonstration to the different softwares and databases which we are going to use in the network pharmacology. So I'll be starting now. So to understand the network pharmacology, we have to know the in which drug discovery pipeline it comes under. So we will firstly take a short look on what's drug discovery. So the drug discovery is the process of finding novel promising drugs and turning them into medicines like that can be used to treat illness, other medical situations is known as drug discovery. And the development. Usually there are multiple steps involved in this procedure. So the identifying a potential targets, like the target identification, our network pharmacology lies in this pipeline in the step of target identification. So the first phase in drug discovery process to identify possible targets for the treatment. This might be particular protein or a molecular link linked to the illness or condition the medication is meant to address. Finding a possible pharmacological target is a crucial stage in the research and development of new drugs. So these are the stages of drug discovery. In stage one, it comes under like target identification, target validation, lead identification, and lead optimization. Then in the second stage, we go wet lab like preclinical development, clinical studies, and drug approval. So what is network pharmacology? The network pharmacology is a field that studies the drug interaction with multiple novel biological targets, moving beyond the traditional one drug and one drug model, one target model. So the key concepts like which in the network pharmacology comes are systems biology integration, which utilizes systems biology to analyze molecular interaction within biological networks. Then the multi-target development. So this is one of the major factor where it focuses on creating drugs that act on multiple targets to address the complex diseases. And then it employs the computational techniques, different bioinformatics tools, the software tools, databases for the data analysis and the target identification. Then another concept is it investigates how combination of drugs can produce greater therapeutic effect than individual, and it is now widely used for the traditional medicine application to identify the uh, molecular mechanisms of traditional medicines. It's now widely used, which validates and enhances the traditional medicines formulations using modern network pharmacological methods. So in general, workflow of target identification using network pharmacology includes the retrieval of genes involved in disease pathology. For these, we use different kind of databases like GeneCards, Uniprot, and we will be looking at GeneCards in this workshop. So then second step is the PPI network construction, which is protein-protein interaction network construction and analysis. For uh, the construction analysis, we use the string database, which produces the PPI network of our input genes, which were came from text mining process, which we will be retrieving from GeneCards, and they are going. Those proteins are going to be analyzed in Cytoscape, which is a software tool. Then for the further processing, like when we got the targets of different of that disease from the network pharmacology, if we go for analysis of those protein, we can use this PDB protein database where we can actually construct the protein 3D structures and download structures from the database. So in the first step is text mining. The text mining is extracting useful information and insights from large volumes of unconstructed text data. It involves employing various techniques such as computational linguistics, algorithms to analyze large number of text data from various sources such as articles, journals, documents, or gene disease association. Sequentially, it refers to the process of mining genes, determination of various associated molecule pathways, and to involve in the protein binding and interaction. So we'll be now looking at the demonstration of one of the database which we are going to use for text mining to mine the genes associated with the disease which we are going to target. So I'll start the demo to GeneCards database. So we are going to use the Google search engine, and we have to just search the GeneCards, which is open access database. So this one is GeneCards database. So this is the interface. We can see there is a search engine where we can put gene or keyword like prostate cancer, collagen metabolism, like any gene we can search here, and we can also search the disease. For example, if I search breast cancer here, so now we can see that we got around 1,60,000 genes associated with the breast cancer. Here we can filter those like protein coding, RNA gene, functional elements, pseudogene, genetic locus, gene clusters, and uncategorizable. We can filter based on our preferences what kind of proteins we are looking at for targeting. The filters applied, now there are only 20,000 genes. So we can export this data into Excel to log in first. So now we can export it in symbols, like mainly we use the Excel. We export this data into Excel sheet, which we can, which will be very useful to handle the data if we export it in Excel sheet. For the demonstration, I have prepared one Excel sheet. I'll show you how those data we can visualize. So these are targets. It's a list of targets we get from GeneCards database. So these are all targets from breast cancer. So this was about the GeneCards, where we can mine the proteins which are involved in the disease, associated with the disease. So in the next step, the gene ontology. We perform the gene ontology for standardize the representations of gene and gene product attributes across species for consistent annotations. In the gene ontology, the components are mainly biological processes, molecular functions, and cellular components. So in the application, it is used for functional enrichment analysis to identify significant biological process or functions or pathways in gene list. The gene list which we are going to put, like now we have extracted genes which are associated with breast cancer. So that comes under this gene list. When we put this gene list into databases of gene ontology, we'll get the enrichment analysis of different biological processes, pathways, molecular function, and cellular components which the genes are proactive. Then in the data integration, it integrates over 1 million experimentally supported annotations from multiple sources. Then mainly this gene ontology databases are publicly available online for researchers to access ontologies and annotation data. Then it enhances the communication and data sharing in genomics and systems biology. So we will be looking at a GeneCards database, which is an open source platform for the gene ontology, enrichment analysis, and cake pathway analysis. So I'll start the demonstration to that. So again, we have to open the Google search engine. These are open source platforms, so they are easily accessible. So this one, GeneCards is 4. This is our database. For demonstration, I'll take few of this list of the genes. We can just paste our list here in text or CSV format. Now I have pasted the. Then we have to select which analysis we are going to proceed with. So I'll for the demonstration I'll put go biological process, gene ontology biological process, and cake pathways analysis, and launch our analysis. It will take few minutes to analyze the data. This is our analysis. We can see it gives us gene count. Means out of our 558 genes, how much are presented in protein phosphorylation biological process, and these are list of genes. It also gives the significance values. Then also relative enrichment it also gives us. These are results for biological processes. Then you can see there is a bar graph down here. For the visualization, we can download this bar graph where color intensity represents the p-value. No, color intensity represents the number of genes, sorry. And this on x-axis we can see there is p-value where they are aligned as per their significance score, where protein phosphorylation is on the top. We can download this table and retrieve this data. So we will see. In the input, we can see that it also performed the cake pathway analysis. This same, the gene count, then significance value, then relative enrichment it has given, and the list of genes which are present in this cake pathways. Again, same. Here, we can see it analyzed the data which we have inputted and performed the enrichment analysis. We can download this table to utilize for our purpose. It will get downloaded in Excel sheet. I have already prepared one Excel to show you how this data is. So this is for Go biological process, where we can see the biological processes and their count, how much genes. For example, we have put the 558 genes in the input. Out of that 558, 126 genes are presented in protein phosphorylation. So it is highly enriched biological process as per our data set. And these are p-value, significance scores, based on how significantly that gene might present in the biological process. And this one's for cake pathway, as we can see pathways in cancer, then lipid atherosclerosis, hepatitis B. Those are highly enriched pathways. And see the enrichment score, then p-values, and the count of the pathways. We can create this data by using the GeneCards software. GeneCards database, which is open source platform for for gene ontology and pathway enrichment analysis. So this was about GeneCards. So now moving further, the protein-protein interaction network. So to build the protein-protein interaction network, we'll be using the string database. The string is a search tool for retrieval of interacting genes or proteins. Is a resource for exploring protein-protein interactions. The data sources, it integrates experimental data, computational predictions, text mining, and curated databases to build the protein-protein interaction network. Then what types of interactions it has? It includes both physical interactions and functional association between protein. Then it covers over 24.5 million proteins from over more than 5,000 organisms. Then in the features, it allows users to create interaction networks, perform functional enrichment analysis, and visualize the protein association with confidence score. In accessibility, it is freely available online for browsing, searching, and downloading the data. And in the application, it is used for understanding cellular mechanisms, identifying therapeutic targets, and facilitating the systems biology studies. String is essential for advancing knowledge in protein interactions and biological processes. Now, we will look at the demonstration for string database. That is search string, and this is the platform. So it's the interface of string database, we can see. So we have to select the multiple proteins, as we are going to put the list of proteins. So this list will come from our gene or our cake pathways or biological processes. For example, we have retrieved the genes from our disease like breast cancer. Then we have put that data in for the gene ontology and enrichment analysis. Now we can select the top highly enriched pathways or biological processes, and from those we can uh, filter out the proteins which are only present in the highly enriched pathways which are involved in our disease. Or based on that, we can select the pathways and biological processes which we are going to target for the future studies. Then we can select the proteins and genes which are only involved in that pathways. So we'll have in the data we'll have that list of proteins. So we will select the multiple proteins option here, and we can just paste our list here. So the demonstration, I'll just paste it from here. For example, these are the proteins we got from the top pathways. We just have to put the list here and then search. Select the organism and select Homo sapien. We are going to so these are some of the list, like similar sound proteins. It's just confirming from us like these are correct or not. We just have to check whatever we are put the data. The queries, it raises some queries like, for example, we have put 10 proteins and the software builds some of the queries like if this is right or wrong or similarly sounding proteins. Like in our list, we didn't filter out our list, so we have these two proteins, so it has given us query. So we have to select one on this. We can proceed further. After checking all these proteins, are they correct or not, we can proceed. So this is our protein-protein interaction network. These are the nodes of the proteins, and these are the edges. You can see the strings are represent the edges. There is like different color codes are there. Like what is the characteristics of the edge? We can like if green we can see from it comes from text mining. Then black is for co-expression. Then blue is for protein homology and experimental determined. Purple nodes are experimental determined. So from what basis or from what source they have established this node, this protein-protein interaction between two proteins. For example, if this one protein is connected to this, and from what basis, from what scientific background, or from which source they got this data, like so we can see that yeah, based on this color codes. Which is the analysis. We can just export this data. And if we send this network to Cytoscape, our further software tool, which we are going to see, where we are going to analyze this protein-protein interaction network. String we are going to use only to build the network, the PPA interaction network. For the analysis, we will be using the Cytoscape database, which will look further. This was all about the string database, where we can put our list of genes and it builds the PPA network. So I'll proceed further to Cytoscape. So the Cytoscape is an open source software platform used for visualizing complex networks and integrating various types of biological data, particularly in bioinformatics and systems biology. So in the key features, it creates visual representations of molecular interaction networks with nodes for molecular edges for interaction. Then it integrates data networks with highly throughput expression data to analyze molecular relationships. Then it enhance functionality through numerous plugins for additional analysis tools and layouts. So in the applications, we can see we can use it to explore protein-protein interaction and gene regulatory networks. Then we can also visualize gene expression changes alongside the interaction networks. And we can also identify active modules within networks related to specific conditions. So we will now start the demonstration to Cytoscape software. We can download this software from Google search engine. It's easily accessible and we can download it from here. So this is the interface of our software. So in the network section, we have to select. It's taking time to start. Yeah, so we have to select the string protein query. So in string database, we have seen that we have to put the list of our genes, and we can take that PIP interaction network to the Cytoscape from that web-based database also. And here in Cytoscape tool, they have also provided this string plugin where we don't have to browse string separately. We can just put that query here, and it retrieves the data from string for the PIP interaction. So we can just have to we just have to put the list here for the proteins which we are going to build the network of a PIP interaction network, and it will already give us the protein interaction network. So we'll put the links. So I'll take this. I have to just fetch the list here. It's loading data from string database. So it has identified 191 genes from our data set. It will take some time to analyze the data. I'll just reduce the number of data for demonstration as it takes time. I'll just take a small list for the demonstration and then run the analysis. Yeah, so this is our PPN network. And for the demonstration, I put the 30 proteins, first 30 proteins. We can put the large library, like large list of proteins. It takes time to analyze the network. So now in the analysis, we will analyze the topology of this the PPI interaction network. Like in the topology, we will be scoring the degree and betweenness values, like different centrality values. So basically, for example, if this GSK3B1 protein we have here, so for example, if we are analyzing the degree value of this protein, so the number of ages it is connected with different proteins, it is interacting with different proteins. It indicates as the signifies as the degree value of that protein, and we can actually analyze this degree value for like to identify. For example, we have the data set of like 100 proteins. From the 100, which is the most potential target, like which is the most interacting protein, which we can take for further analysis. We identify from these 100 proteins, we can identify the protein which is highly interacting and which can be the potential target in the disease, which plays the major role in the disease or that specific pathway from which we have taken the data from. We can see GSK, like it's 1, 2, 3, like around 10 to 12 is going to be its degree value. So for this analysis, like this is small pp interaction network, so we can see the network and see the edges. For the large, we can't analyze it through visualization. So for that purpose, there is a plugin called as Cytoscape 2.2. This is one. Cytoscape 2.2, which we have to download in this Cytoscape. So I'll just this is tab for Cytoscape 2.2, where we can select these different centralities. Like for example, as I discussed, this one is degree value. This one betweenness. Betweenness is, for example, this protein is interacting with two proteins, so its degree value is 2. And for example, it's interacting with this one and this one. For example, and this protein is interacting with another 2, and this is with another 10. So number of the degree values of interacting proteins are also get calculated to this protein. For example, its degree value will be 2, and the betweenness value is going to be somewhere around 14, 13 or something. We will look the data like after analysis how it works. So we have to select the undirected network. If you select directed, we have to select like, if for example I have selected the 4 proteins here which I find more interacting, and select the directed option, it will only analyze these 5 proteins only. So we'll select the undirected as we have to analyze all the proteins and start the analysis. So it has analyzed degree and betweenness value. We can see here the betweenness, undirected, and degree values of the proteins. We can export this data in the CSV file. It get exported. We can convert it into Excel too, and we can analyze the data. For the demonstration, I already prepared one Excel sheet. I'll show you how that data looks. So this is how we get the data for degree and betweenness values for the proteins. So these are the proteins which are analyzed in Cytoscape, and these are degree betweenness values. We can just apply the filter here. Like if we apply the filter, I already applied filter, and then yes, and we can select the largest to smallest. It will rank the proteins based on degree values. So we can see that AKT1 is a highly interacting protein among the list. So I have selected these 10 proteins, which we can use for further analysis for the validation purpose. So this was about cytoscape, where we can analyze protein-protein interaction network, and from the analysis of protein interaction network, we can find out the potential targets, like key targets which are highly interacting, which are playing the role in disease pathology. So this was about network pharmacology. So how we can proceed further after network pharmacology? So we can go for target validation, then exploration of traditional medicine, then personalized medicine approach, then for the clinical trial, then drug design optimization, then integration of this data with omics data, where we can clinically validate the targets which came from the network pharmacology. So this was our network pharmacology. So I'll just give a demonstration of molecular docking, where I'll select the protein like AKT1 and select a ligand for the demonstration, and I'll be performing the molecular docking. I'll deliver this the second part of this workshop, like I'll show the demonstration of molecular docking. So the molecular docking is a computational method used to predict the preferred orientation of one molecule, like ligand, to another receptor when they form a stable complex, mimics the process of ligand binding to a protein target. And why it is important that it accelerates the drug discovery process, reduces cost associated with experimental screening, and provide insights into protein ligand interaction. Now, the reason to associate the molecular docking to the network pharmacology is to we can in silico validate the computationally validate our proteins which came from the network pharmacology. Like for example, if we got 10 targets, we can further take those 10 targets and take different types of ligands which came from plants, or any novel ligand which we are finding the target for, which we can take that and analyze if which protein is based on the binding score. We can analyze which protein is giving the most significant binding affinity. So the first step in the molecular docking is target preparation, where we have to retrieve the 3D protein structures. For that purpose, we will be using the PDB database, which is Protein Data Bank. It's a open access database for 3D protein structures. So I'll give the demonstration how to download the protein structure. It's open source database. So this is the interface of PDB database. You can see here we have to search the terms like protein ID, like PDB ID, and we can just put, for example, the highly interacting protein which we got is AKT1. You can search the active one. Yeah. So these are the protein structures which are available in the database. Based on this code, we can select where we can see the macromolecules. We are seeing the potassium channel AKT1, potassium channel AKT. We can select. We can also, there are some filters are available where we can select the source of organism, then experimental, if they are experimental or not. We can apply these filters and then search. For example, if I select the experimental, it's showing me 8 proteins. And we can just download this protein from here. For example, if I have to download this protein, I'll go to download files and I have to select this legacy PDB format. And protein will be downloaded in this PDB format. So this is about the PDB database where we can retrieve the 3D structures of proteins. And now in the second step of target preparation, where we have to clean the protein structure. For example, if I open the structure in Discovery Bio, so the Discovery Bio Studio is another software which is a open access software where we can visualize the protein structures, and we can also do modifications, and we can clean the protein, and we can also in the last step of docking where we can see we can analyze and visualize the molecular docking interactions. For example, like I open this structure in so now we can see these red dots are water molecules which we don't need anything. If we press the Control H, the hierarchy bar will get opened here, like hierarchy tab. Then we can see the different components in this protein, like these chains, like protein chains and het atom, then water molecules, then what are the active sites and the protein groups. So we will be only needing the protein groups. If we select the het atom, so all the water molecule and unwanted which are not part of the core skeleton of protein are selected in this het atom option, where we can cut this. So now all the het atoms are deleted, and now this protein is ready for the further processing. So by this process we can clean our protein. So we are done with the target preparation. Proceeding further. So the second step is ligand preparation. We have prepared our target, like protein target, and now we will prepare the ligand, which we are going to bind with the target. So for the ligand preparation, we use the PubChem database. So we can use the PubChem database. There are different databases like Zinc and all. So we can retrieve the 3D structures of the chemical entities which are leads or any chemical compound which is under investigation as a ligand. So in the ligand preparation, I'll show one demonstration to this PubChem database. So it is open source platform. It's easily accessible. This yeah, so this is the interface of the software where we can see the search engine where we can search our ligand. For example, I'm taking camphorol as our ligand, which is a flavonoid from plant. For example, this is our drug under investigation. We are finding the activity of this drug or ligand or lead or heat, we can say, for our disease treatment. We can select this ligand. We have to just download from the download option. There are different options available where we have to download the 3D conformer in SDF format. So SDF format is accessible to the Pyrex software. So we will be using this SDF, which is structured data file format, and we can just download from here. So now it's downloaded. So CID number and conformer. We have to save it in our workplace, and we can utilize this for the docking. So this was about the ligand preparation, where we have to just download the 3D structure of our ligand from PubChem database or any database we can use. For the demonstration in this workshop, we are looking at PubChem database. So I'll proceed further in molecular docking. So for the docking, we will be using the Pyrex software, which is open source software tool designed for virtual screening and computational discovery. The Pyrex utilizes the Autodock Vina engine for the docking, which is well-known engine for docking, where we can run multiple ligands at same time. One protein, multiple ligands, we can see the binding affinities to it. Now we will directly go to the demonstration. Well, you get the idea how this software works, and you can perform docking easily using this Pyrex software. This is easily available software we can download from Chrome actually. I have already downloaded from PC. So this is the interface of software. It's so user-friendly software, and there are so many features which we can utilize for the docking. And in the first, now we will load the protein or target which we are going to use for docking. So load the protein, go in the file, and select the option load molecule. So here I created one shortcut. So this is our PDB file. So we can only see this PDB file. So we have to open this. So this is protein file. I've just opened in Irix software. So we have to just convert it in orthodox format. So I'll select the option make macromolecule. You have to just right click in the molecular tab. We can visualize our protein here. We can also visualize different amino acids. We can see here. So we have to just select this entity, then right click on it. We'll see this option save as PDB, remove from scene, display, and autodoc, where we have to select the option autodoc, and we have to make it macromolecule. Macromolecule means it will act as a target, converted in macromolecule. So in the autodoc tab in macromolecule section, we can see the 4-region AKT1. 4-region is PDB ID of this AKT1 target. So now we'll load the ligand in the software. So to load the ligand, we have to use the OpenBabel plugin here. So OpenBabel is used to convert the different types of file formats to autodoc, like any format. Like we can convert HDF to PDB, we can convert HDF to PDB-QT, which is used for autodoc format. So we'll directly open our structure in this OpenBabel format and then convert it into PDB-QT format, which is suitable for the autodoc function. So in the right side, we can see the insert new item. I can just load this camphor which we have downloaded from the PubChem database. This is which is a SDF file. So now we are down. So this structure of our camphor, and in this tab, you have to just right click and minimize the energy, like torsion energy of the ligand, which will be useful for the docking. We will select the minimize selected. Okay, you have to select one algorithm here, force field, which we have to apply, and select UFF force field. By this method, we can also convert 2D structures in 3D into 3D conformers using this minimization of energy. Then we have to convert it into orthodoc ligand format, which is PDB-QT format, which is suitable for the orthodoc engine for the docking. And convert it into orthodoc. It has converted. We have to just refresh. We can see our ligand here. So now for the docking, we have to open the Vina wizard, which is available. There are two engines they are provided. One is Vina and Autodoc. We are going to use Vina, where we can select the multiple ligands for the docking. Now in the select molecules tab, we can see the no ligand selected, and this one is already selected. It's 4-region PDB-QT. We can see. Now to select the ligand, we have to click here. If there are multiple ligands, we have to just control and click all the ligands which we are going to dock. Then we have to select the run winner. So the second step, we have to adjust the grid. So in the auto grid, we can see a special box here, cube, where we have to adjust this cube where our binding site is. For example, if I go in the molecule side, for the protein, like if I take the well-known proteins which are already been established target for diseases, there is data available that what is the binding site on that macromolecule structure. For example, here is one option called as toggle selection, where it highlights the amino acids from the, for example, if I select this ILE val for the demonstration, we can see it is highlighted in our 3D space. Yeah. So for example, from the literature and all, from the experimental studies, it is established that AKT1 has this target site where these 5 amino acids are involved. Where I can see this is now my binding site. I can adjust the grid around this and proceed further with the docking. For the demonstration, I'll be not selecting a specific binding site. I'll take whole protein for the and just maximize it. Like my whole protein will now act as a binding site, where will be wherever the ligand is more suitable to bind, it will bind to this protein, and we can get the binding affinity for the ligand protein interaction. So I've selected the protein, I adjusted the grid. Now I'll run the docking. We can see multiple steps it's taking, like CTM scoring function, analyze. Now it's analyzing the binding site. Uh, Docking is done for the AKT1 and camphorol. So we can see in this pocket, the ligand is bind with certain amino acids. You can visualize in 3D space. These are different binding affinities like RMSD based on root mean square divisions, where zero value represents the what is the best position for the docking, where we can see the highest binding and affinity. The smaller the binding affinity, we can see their negative scores. Smaller the binding affinity, which shows the strong bonding interactions, which indicates a strong bonding interaction. So it is good when it shows the lowest digit of smallest binding affinity, where we can see minus 9.3, the smallest binding affinity in kilocal per mole. So it is on top, which is the binding affinity of camphorol with AKT1 as per our analysis. So in the further step, if we have to visualize this properly, we can visualize this properly in Pyrex software. We'll be using again Discovery Biovia, as I already discussed that it is used to visualize and analyze the molecular binding 3D structures of the binding, where I have to just convert this binding scene into PDB format, and then we can visualize and analyze our interaction in uh, Discovery Bioware Studio software. So I'll just show you how that works. So this is our overall scene, which we can see it here, which is for minus 9.3 binding affinity. This is our interaction scene, which we can convert into PDB, where we can save it as PDB. Save it in our workshop. So this is our interaction scene. I've saved it in file. So now we will proceed further for visualization and analysis of our ligand protein interaction. This is our interaction file, where we can open it with Discovery Biotech Studio. We are opening this file in Discovery Studio. We will visualize and analyze the protein. I'll show you how it works. So this is our ligand, which is, we can't see the protein here. So as it aligned in the way that it is going to bind the protein. So we have to just open the protein here and then paste this. This is not just the ligand we are seeing. It's the loci of this protein. This location of the ligand on this protein, on the protein. We have just paste this, copy this ligand and paste it into the our protein file. So I'll just open the protein file, which we have used for docking. The file, I'll just open the protein. This is the protein file. Control C, I'll copy this protein and paste it here. So now we can see it's binded in this pocket. So here is this tool tab where we can have to select this tab, receptor ligand interaction. Then we can see this interface here in the left side, where we can do many things with this. We can modify the structure and we can analyze in different ways. Like now I can see I have to just, I'll just control open the hierarchy where I can select this ligand and define it as ligand. Now Scamferolic 81 is now defined ligand for the visualization. In the ligand interactions, we can see how ligand is interacting with different amino acids in the protein. Now I can see that in 2D diagram, we will get to know on which amino acid it is binding, like how the structural entities of ligand are interacting with different amino acids present in the protein. We can see this. These are aspartate, threonine, different types of amino acids which are present in the AKT1 are interacting with our ligand. And these colors of the bonds represents the type of bond. Actually, we can see conventional hydrogen bond and donor-donor bond, PNN bond, pi-pi stack bond, and pi-alpha bond, where we can visualize our interaction. If we have to analyze this, we have to look for the characteristics of our bond. We have to select this non-bond tab. I'll just raise it up. Now you can see how the oxygen from the ligand and this amino acid from our macromolecule. Now it is one of the interaction from our docking. So we can see different parameters like what is bond distance, and what is the category of that bond, then what is type of bond it is establishing with the amino acid, and from which amino acid to which group of our ligand, like oxygen to this serotonin of our protein, it is interacting by conventional hydrogen bond. Then we can different parameters, structural parameters of the bonding like angle, bond angle, then these are different chemical parameters where we can see, analyze our binding interaction. So this was about analysis, how we can analyze our ligand protein binding using the Discovery Bio Studio. So this was about the molecular docking and the analysis of that docking interaction. So we looked into how this analysis works now. So that was all about the network pharmacology and the molecular docking. We have shortly discussed how, shortly introduced, looked at the introduction to the different databases, software tools used in this process. And that was all from my side. I have covered so many parameters and different aspects of network pharmacology and molecular docking and represented the demonstration for the same. If there are any questions, I'm ready to answer.
Thank you so much, Mr. Harshad, for sharing your valuable expertise with us today. Now we will open for the floor for question from the audience. Our first question is from the audience. How can network pharmacology be applied to repurpose existing drugs for new indication?
Like for the repurposing, for example, if there is a drug marketed, like we can see aspirin or anything, which is already marketed drug. So by the network pharmacology approach, we can identify different targets. Like we have to check the activity of specific drug. For example, routine is there. Routine is like there is so many literature published for routine, like it is active in different types of diseases. For example, we have to check the activity of that routine in the specific disease. For example, if I chose the breast cancer, now I want to check the active routine of one breast cancer. For example, routine is never been explored for the breast cancer. So now I can just identify different targets which are associated with the breast cancer and check the activity of routine if there is any possibility that it is targeting different proteins or genes which are involved in the breast cancer, and we check the activity which will be useful for the repurposing of drug in that pipeline of drug discovery. We can use network pharmacology.
Our next question is, how can network pharmacology analyze the safety and adverse effect of drug?
So in the network pharmacology, not just diseases, we can retrieve the targets for different conditions like toxicity, or for example, there are different adverse events. As we can see, we can just select those and for example, I'll just show you. If I go to GeneCards, not just disease, if I select any condition, if you can see, we'll see nausea is there. Yeah. So nausea is one of the side effects. So we can take all these targets and analyze it using network pharmacology. We'll select the specific targets and drug for which we are going to identify the its role in this side effect. We can actually use it for that purpose only. So not just disease, we can also target the side effect if it's active on that. Our drug is playing a role in that side effect or not. We can identify that.
Okay, our next question is, what are the primary challenges in network pharmacology research?
Primary challenge is like it is limited to the databases actually. Whatever the data is already been published or available in public domain, which we can use. The experimental data which yet to be analyzed, or we can see yet to be explored, we can't go there. The data which has been already published or available in those softwares only we can analyze. So that's the main, like, primary limitation of network pharmacology. And as we go further, it is like minimizing so many steps, but at the end we will be needing wet lab procedures for the validation and all. So that's limited that too, which is the basic limitation of network pharmacology.
Okay, our next question is how can machine learning and AI improve predictive accuracy in network pharmacology models?
AI we can use actually like different prompt engineering techniques we can apply in network pharmacology, which can accelerate the process. Like in the process of text mining, we can see there we have to search different literatures and all. By the prompt engineering, we can minimize those steps and we can accelerate this process. So we can analyze the large number of data at the same time by using AI.
Okay, our next question is how can knocking results be validated experimentally?
In the docking results, we are getting the binding affinities for the different ligand protein interaction. For example, in the plant, if I selected 10 of proteins and 10 proteins in the disease and 10 ligands, we can run the scoring function for all and we can rank based on docking score. Like we can filter out one ligand, one protein, or one ligand, different types of proteins, and those data we can use to design our preclinical study like cell-based assays or any like for the validation of protein, like different type of cell-based assays, and we can also design the animal studies based on that.
Okay, our next question is what was the p-value in gene ontology?
P-value significance score, like p-value indicates that how significantly that protein or the list of protein is present in that like any biological process or pathway. It's probability value, like it shows the significance, like that protein is present there by chance or how much chance is like our null hypothesis can be rejected there.
Okay, what type of data are integrated in network pharmacology analysis?
Data from experimental studies, data from different public domain databases and literatures, different predictive data also, predictive algorithms also used in, and these all are integrated in network pharmacology.
Okay, our next question is beyond any drug discovery, how is network pharmacology being applied in personalized medicine?
Sorry, what is question? Which medicine you said?
Hello?
Yeah, which
Beyond drug discovery, how is network pharmacology being applied in personalized medicine?
in personal medicine? Yeah, okay. So in personal medicine, like we're getting different, like large library of targets, large library of biomarkers, which we can target in uh, network pharmacology. Like for example, if in the disease in chronic conditions like cancer, where we can see that there is need of personalized medicine because different person are showing different biomarkers in disease, like different types of genes are expressing in it. So based on the libraries we can select those proteins or set of proteins. Based on that we can select the combination of ligands and like optimize those, like which protein, which ligand, which we are going to do, and based on that we can prepare the personalized formulations for the disease conditions.
Okay, our next question is through network pharmacology, can we study drug resistance studies and how?
Drug resistance studies. uh, in drug resistance, as we can see, the if for example we have the different, like in pharmacology, we can analyze multiple drugs at same time. So in drug resistance, we can see if one drug is not acting on that target, the other is acting. So we can minimize the drug resistance, also study the drug resistance, like different proteins are there which are responsible for drug resistance. We can analyze those in pharmacology too and select the target based
Okay.
resistance to the drug.
Our next and last question is, how does network pharmacology aid in identifying biomarkers for disease prognosis and treatment response?
Like neuropharmacology, can you repeat the question? Sorry.
How does network pharmacology aid in identifying biomarkers for disease prognosis and treatment response?
We can only identify the targets based on, like we can go ligand based approach or disease based approach where we can identify different type targets, and those targets are only act as biomarkers. So in further
Okay. Thank you so much for your detailed explanation. Now let us move to our next audience question. So thank you, Mr. Harshad, for your answering all the questions and clearing our doubts. Thank you so much.
thank you everyone. Thank you Bhavesh, and thank you Swalife team for giving me this opportunity to deliver the workshop on network pharmacology and talking. I hope I did well, and everybody understood how the network pharmacology and molecular docking works, and we can apply this in drug discovery process for the target identification and the validation. Thank you very much.
Thank you.
For further, I would like to introduce Komal Vanare, a dedicated postgraduate student in pharmacology. She will be enlightening us today on the fascinating topic of Swiss ADME, pharmacophore modeling, and ALVA QSAR, which are powerful computational tools in modern drug discovery and development.
Thank you, Sakshi. I would like to thank you, Swalife Biotech, to give a chance to me to give a workshop on network pharmacology. I am sharing my screen. Is the screen is visible, Sakshi?
Yes, the screen is visible.
Okay. Just a minute. Okay.
Your voice is not coming, Komal.
Hello? I'm audible?
Yes.
Hello? Okay. Good morning, everyone. Today, I will be discussing some essential tools and methodologies used in the drug discovery process. We will be covering topics like drug likeness, prediction, pharmacophore modeling, and QSAR, also live demo of these tools. Let's start with the Swiss ADME, which is an excellent online tool used to predict the drug likeness of small molecules. It helps us understand two ADMET properties: absorption, distribution, metabolism, excretion, and toxicity of the compound. By using this tool, we can evaluate how a molecule will behave in the human body. Also, this tool checks the several important properties such as a Lipinski rule of 5, solubility and permeability. Also, the cytochrome P450 interaction, cytotoxicity prediction. Also, in application in drug discovery, it favorable pharmacokinetic profiles. Swiss ADME is very useful during the early stages of the drug development. It helps us identify the promising drug candidate that have good pharmacokinetic profiles, which means they are likely to behave well in the human body. And it also aids in the preclinical screening, helping us avoid the costly failures. This is the workflow of the Swiss ADME. Firstly, go to the PubChem, search your molecule, then copy the smiley and paste into the Swiss ADME. Instead, you draw the structure also, then run and analyze the result. After that, interpreting the Swiss ADME result. In which once we get the result from Swiss ADME, we interpret them like a Lipinski rule of 5. This rule helps us to evaluate a molecule's drug likeness based on factors like its molecular weight, lipophilicity, and number of hydrogen bond donors and acceptors. Along with this Lipinski rule, Swiss ADME also predict the ADMET properties, how the compound will absorb, distributed, and metabolize, and its potential toxicity. In for the potential drug-drug interaction during a metabolism. Insolubility and permeability, we check if the compound is likely to be absorbed well in the body, and the positive and negative outcome of the compound passes the test. It is good candidate for the further development. If it is fails like having a poor solubility or high toxicity, we may need to modify the compound. Now, let's move to live demo of Swiss ADME. Firstly, go to the PubChem. Search your molecule. Search your molecule. I will take azadiractin as example. Copy the smiley. Then go to the Swiss ADME website.
Komal, you
Yes,
are
sir.
sharing only the PPT. Your screen is not visible.
Okay, sir. Okay. Now visible, sir?
Yes, visible common.
Okay. First go to the PubChem. Then search your molecule. I will take example of azadiractin. Then copy the smiley. Then go to the Swiss ADME website. paste the smiley. Instead of, you can draw the structure also. Then run. Wait for few minutes. The result is loading. Wait for few minutes. Here result is coming. The molecule physiochemical properties, its lipophilicity, water solubility, pharmacokinetic properties of the compound, drug likeness, and medicinal chemistry. These properties are coming. You can use these properties for the lead optimization of this molecule. After that, pharmacophore modeling, the blueprint for drug design. Let's now talk about the pharmacophore modeling. This is the computational technique that helps us identify the key features of a molecule that are needed to interact with a specific biological target. These features might include the hydrogen bond, donors, hydrophobic areas, and aromatic rings, all of which are critical for a drug binding. Pharmit also used for the lead compound identification. Also, workflow of the Pharmit. Go to the protein database, find the protein target that we want to work with. Once you find your protein of interest, copy the protein code from the PDB, then paste into the Pharmit, submit, and search the molprot. Adjust the feature that give the low RSMD value. Once you get a low RSMD value molecule, go to the PubChem link and take that synthetic molecule or a lead molecule for the comparison with your herbal molecule or for further study. Interpreting pharmacophore models. These are the pharmacophore features like hydrogen bond donors, acceptor, hydrophobic region, just to get a low RSMT value molecule. Once we have a pharmacophore model, we can use the virtual screening to compare a large compound libraries and find the best candidate. We can also optimize existing compound to improve their binding affinity and specificity by adjusting them to better match the pharmacophore. We can show the demo of Pharmit. Firstly, go to the protein data bank. Find the protein target. You can search your protein. I will take a SOD1. Then take a 3D structure of protein. I will take this. Then copy the protein code 6FN8. Then go to the Pharmit website. Search here your protein 6-FNet, then submit. Add here your pharmacophore features like uh, hydrophobic, uh, negative ion. You adjust, then search molprot. Here the pharmacophore results come. Wait for some time. Here are some molecules are come. You can adjust the features for reduce the low RSMD value. You can adjust these features on off. Search molprot. Here 0 value is coming. Go to this link. Here we get the lead compound. You can copy the smiley of this compound and paste into Swiss ADME for the lead optimization. Also paste into QSAR for the lead optimization. You can also compare this molecule, your synthetic molecule with herbal molecule also. We take an example, copy that smiley, go to the Swiss ADME. Paste, run. Here the synthetic compound lead compound properties are coming. You can compare with this herbal molecule also. After that, ALVA QSAR predicting the biological activity from molecular structure. ALVA QSAR is a web-based tool designed to build the quantitative structure activity relationship models which predict the biological activity of a compound based on their molecular structure. Application in drug discovery like activity prediction for a new unknown compound help to prioritize lead compounds by predicting their biological effect, assist in identifying the structure activity relationship to guide the further optimization. It is used to for the lead optimization. This is the workflow of ALVA QSR, in which firstly copy the smiley, or instead of you can draw the structure, then predict the biomagnesia factors, PMF, also like the Daphnia magna LC50, also bioconstriction factor, biodegradability factor, also the fathead mino LC50 factor for the toxicity study. This model predict the biological activity of a new compound, and we need to interpret this result, check the evaluation matrices to access the model accuracy and predictive power. We show the demo of go to the ALVA QSAR website. You need to log in the ALVA QSAR firstly. Then go to ALVA QSAR. You have to enter the smiley, then go to Pubchem. You can copy this smiley. Firstly, you have to copy the smiley. Of the draw molecule, instead of you also draw a structure, draw a molecule structure, then paste the smiley. Then you can predict this biomagnetism factor, Daphnia magna, bioconstriction factor, biodegradability, Fatedmino. The biomagnetism factor for this measure helps to access how a compound accumulate and magnify through the food chain. It tells us potential for a compound to accumulate in organism and higher tropic levels. Also, the Daphnia magna LC50 that used for the toxicity testing of the compound. LC50 refers to the concentration at 50% of the test organism dye. Also predict the bioconstriction factor, like it helps us understand how a pollutant might enter the food chain through the water sources. Also predict the biodegradability, breakdown of the compound. Then this Fatedmino for the toxicity testing of the compound. This these all are used for the lead optimization of the compound. Biomagnetism factor BMF, predict, wait for some time, calculate prediction. These are the information about the compound you can use for the lead optimization. You can also download the report. Also, you can predict like a properties Daphnia magna for the toxicity testing of the compound. These are the information of the compound. You can also download this report. These are the demo of ALVA QSAR. This is all about the Swiss ADME Farmit ALVA QSAR. This is from my side. I have covered the demo of these tools. Thank you.
Thank you, Komal, for delivering such an insightful and informative lecture on Swiss ADME, Pharmacopore, and ALVA QSAR. Your session has truly understanding of how the computational tools can accelerate drug discovery and help in developing better therapeutic strategy. There are some questions from our audience. First question, what is the use of the Swiss ADME?
Yes, use of Swiss ADME for the drug likeness prediction, ADME prediction, adsorption, distribution, metabolism, excretion. Also for the lead optimization, also Swiss ADME is used. Also the physiochemical profiling, like medicinal chemistry filters, and for the virtual screening, also Swiss ADME use. Hello.
Okay. Our next question, what challenges do researchers face when interpreting results from Swiss ADME?
What, Sakshi?
What challenges do researchers face when interpreting results from Swiss ADME?
Mm. Swiss ADME uh, ADME uses established models like Lipinski rule of five and other ADME prediction. And these models are based on a general assumption about the molecular properties and may not always predict the outcome accurately for every compound. And researchers should avoid the relying for the prediction and should verify them with the experimental data whenever possible.
Okay,
Hello?
our last question. What is the role of QSAR format in drug discovery process?
QSR is used for the lead optimization. You can optimize your lead with QSR. You can predict your biomagnetic properties like biomagnification factor like a Daphnia magna. Also, the compound toxicity testing also done with QSR.
Okay, thank you, Komal, for patiently answering all the questions and clearing our doubt. Thank you so much for this session.
Yes. Thank you, Sakshi. Thank you, Swalife Biotech, and thank you, Dr. Praveen Bade sir, to give a chance to give me to take a workshop on the network pharmacology. Thank you so much.
Thank you, Komal. Now we will move forward to the next segment of the day. Good morning. The next segment of the day, the poster presentation session. This is a wonderful platform where young researchers and academicians showcase their innovative work, highlighting new perspective that can shape the future of drug discovery and pharmaceutical science. It's my privilege to introduce our first set of presenters for these existing sessions. Our very first presenter is Ms. Shreya Abhay Firke, a PG student from Bharati Vidyapeeth College of Pharmacy, Navi Mumbai. She will presenting on a highly relevant topic, nanotechnology enabled drug delivery, emerging platforms, challenges, and future perspective. Her research is guided by Dr. Viniti V. Khanvilkar. Let's welcome Ms. Shreya Abhay Firke.
Can I start, sir?
Shreya, you can start now.
Okay, sir. Is my screen visible? Hello? Hello? Am I audible, sir?
Your screen is not visible.
Okay, sir. Just a second. Is it now, sir? Hello.
Still, your screen is not visible. It's in the process. Yeah, now it is visible.
Okay, sir. Thank you. So today, myself Shri from Bharati Vidyas College of Pharmacy, I am going to present a poster, Nanotechnology and Drug Delivery, Emerging Platforms, Challenges, Future Perspectives. Imagine a world where medicines can seek out disease cells just like guided missiles, delivering healing power wherever needed with minimal side effects. So here comes the nanotechnology in picture. Let's understand first what nanotechnology is. Nanotechnology involves science and engineering of manipulating the matter at a nanoscale, which is from 1 to 100 nanometers. At this scale, materials exhibit unique physical, biological, and chemical properties that differ from their bulk counterparts. In drug delivery, nanotechnology allows precise drug delivery at the disease sites, minimizing side effects. So, what is
Hello?
hello? Is my screen visible, sir?
Your screen is not visible. Your voice is also not that clear.
Shreya, you can share your
Am
screen.
I audible now? Sorry.
Shreya, you can share your screen.
Okay, sir. Is my screen visible now?
No, nothing.
Is my screen visible now?
Yes, your screen is visible.
Thank you. So, what is the need for nanotechnology enabled drug delivery? The necessity arises from several key advantages that are overcome over shortcomings of the traditional methods. The first is targeted delivery, which ensures drug releases to the specific sites and tissues via reducing the off-target effect. Second is enhanced bioavailability, which improves absorption and stability of drugs, particularly hydrophobic compounds that are poorly soluble in water. The third one is controlled release. Nanotechnology allows sustained drug liberation over time, maintaining therapeutic levels without frequent dosing. Fourth is lower dosage. Minimizing the dosage ensures reduced toxicity by concentrating drug at the site of action. The last one is versatility, which enables customization for the formulations available in the market are
Shreya, your screen.
Donoxone, Calyx, Pezem.
Shreya, you can share your screen.
Yes, sir. Sir, is it visible? Hello?
Yes, you can start, Shreya.
So, the formulations available in the market are Donoxone, Calyx, Pezenir, Abraxane, Rebinine, Rapune, Ovinide, and Myoset. The first nano drug was Doxil, which is liposomal doxorubicin, which was first FDA approved drug in 1995 for the treatment of sarcoma and ovarian cancer. The market insights include the market insights. The global nanotechnology drug delivery market is experiencing rapid growth, estimated at USD of 108.08 billion, which is in the year 2025. This is driven by chronic diseases, aging population, and investments in the R&D. Projections indicate compound annual growth rate, that is CAGR, of 10 to 12% through 2032, potentially reaching an USD of 228 billion. North America holds the largest share due to robust infrastructure and companies like Pfizer and Johnson & Johnson. The emerging platforms here are hydrogel, which is a 3D network of hydrophobic polymers that swim in water. Hence, they are ideal for sustained release. The example for this is PEG-based hydrogels, which are used for wound healing and encapsulating antibiotic to prevent infection. Micelles. Micelles are self-assembled amphiphilic structures with hydrophobic cores for drug loading and hydrophilic shells to ensure their stability. Here, Genexol PM, which is a micellar paclitaxel, is used for treatment of cancer. Dendrimer. Dendrimers are branch tree-like polymers with precise molecular architecture, allowing high drug loading and targeting via surface modifications. Here, the example is delivered siRNA for the gene therapy. Nanoparticles can be polymeric, metallic, and inorganic. For polymeric, PLGA-based are used in the vaccine delivery. In metallic, gold nanoparticles are formulated for photothermal therapy in cancer. Inorganic, here, silica for image-guided delivery is used. Carbon-based, here, first is carbon nanotubes, which penetrate the cell membranes. Graphene and graphene oxides are used for large surface area absorption, while carbon nanohorns are used for photodynamic therapy. Fullerenes are used for antioxidant are used as antioxidants in and neuroprotection. Carbon nanodots are used for bioimaging, while carbon nanodiamonds are used in sustained release in the orthopedics. Liposomes are traditionally used counterparts, which are phospholipid vesicles. They are of the three. They are classified into three basic types: multilamellar vesicles, a large unilamellar vesicles, and small unilamellar vesicles. The mechanism involved in targeting are active targeting, passive targeting, and stimuli responsive targeting. Active targeting functionalizes nanoparticles with ligands wherein the ligands bind to the specific receptors on the target sites. So here the example is trastuzumab, which is conjugated liposomes that target the HER2 cells for the breast cancer. The passive targeting is EPR, that is the permeation and retention effect, where nanoparticles extravasate through the leaky vesicles in the tumors, thereby accumulating in them and reducing the effect of the tumor. Here the example is Ebrexane, which is a marketed formulation which relies on the accumulation in the solid tumors for pancreatic cancer treatment. The stimuli responsive mechanism is a mechanism in which the drug liberation is based on the response to environmental cues like pH, temperature, enzymes, etc. Challenges. Despite the promise, challenges persist. Scalability involves high costs, complexity in large-scale production with yield inconsistencies due to, in case of dendrimer synthesis. The biocompatibility is mainly due to the biocompatibility as a challenge is mainly due to the immune responses and toxicity. Regulatory approval as a challenge is because continuous and rigorous safety data is required, and which is delayed, and hence only a fraction of candidates reach the market. Clinical translation. Lab success does not actually mean that the drug is safe, and which hence rigorous clinical trials should be done and patient outcome should be monitored. But this patient outcome is variable due to biological heterogeneity. The next is environmental impact. Nanoparticle waste and ecological toxicity. Also, the safe disposal of nanoparticle waste which are generated during the synthesis are the major challenges which include the point environmental impact. Even though there are many challenges, there are also future perspectives which can help us overcome these challenges. The first one is smart nanoparticles, which incorporate sensors and feedback loops for real-time response monitoring of the disease. Green nanomedicine. Green nanomedicine is eco-friendly synthesis using green solvents and biodegradable carriers so that sustainable production process is achieved. Multifunctional hybrids. Multifunctional hybrids is today the latest booming technology used. Here, a combination of diagnosing, imaging, and therapeutic function is made in one. So the term used here is theranostic, that is therapy along with the diagnostics. The best example here is magnetic liposomes for MRI guided imaging. The next point is AI integration.
Now you
Here,
have 2 minutes left.
sorry, sir.
You have now 2 minutes left. Continue.
Okay, sir, thank you. AI integration, which optimizes designs via machine learning. The CRISPR technology is used via nanovectors, which promises gene editing for the gene delivery. Personalized and precision therapy, which tailors therapies to individual genetic and molecular profile, enhancing the patient outcome, which is hampered due to biological heterogeneity. Enhanced translation, which is achieved through streamlined clinical trials. Here the data is AI is reducing development by 30%. It is reducing the development time by 30%, and CRISPR nano delivery is achieving 80% gene editing efficiency. The conclusion is, hence, nanotechnology is transforming healthcare by enabling targeted, efficient, and patient-centric healthcare. Patient-centric drug delivery with innovative and sustainable approaches. Thank you.
Thank you so much, Shreya.
Sorry for the technical glitches.
Yeah, it's okay. Thank you so much for that very insightful presentation. Now we open the floor for the questions. Respected judge and audience, kindly feel free to ask your questions.
There are a couple of the challenges you're mentioning about the clinical translation. And in the future
Yes.
perspective, you are also talking about enhanced clinical translation. So what is the difference?
Sir, regarding the clinical translation, although we get what I was saying is, although the lab success is achieved, it doesn't actually mean that clinical benefit will also be achieved. Hence, clinical trials should be performed should be based on the robust methods, while streamlined clinical trials should be performed so as to overcome this challenge, uh, following a well developed method, so that regulatory approvals in future doesn't create a problem here.
So what type of regulatory approval problems are there in nano products?
Sir, the main regulatory problems include the biocompatibility. Uh, the uh, the the biocompatibility is due to the biological heterogeneity which exists person by person. So not just one drug will show same effect on both the people. Also, side effects may vary from person to person. So all these things should be considered uh, for for considering for formulating a formulation which can give which can achieve good regulatory approval.
Can you give me an example of a nano formulation which is registered in India recently for cancer?
Sir, for cancer, it is Calyx, which is used for breast cancer.
Helix. From which company it is and when was it registered?
Sorry, sir, I don't know that.
Okay, thank you. Thank you, Shreya.
Thank you, sir.
Shreya, the question is from the viewers. The first question is, how does nanotechnology enhance the bioavailability and controlled release of drug?
Okay, so regarding the bioavailability, the emerging platforms here are liposomes, which are phospholipids. So, liposomes which are phospholipid vehicles, so so these drugs can be encapsulated into these nanovesicles. Hence hence they can reach the targeted site and bioavailability can be achieved. Regarding the controlled release, polymeric nanoparticles can be formulated so that the release can be controlled using a polymer. So if we want immediate release, we can adjust the polymer accordingly.
Yeah, the next question is, next and last question is, how can nanotechnology be adapted for personalized and precision medicines?
Okay, regarding the personalized and precision therapy, the individual genetic and molecular profiles can be studied for that person, and hence by studying that profiles we can adjust, simultaneously we can study our formulation aspects, and using integration of AI we can personalize that therapy for that person based on that.
Okay, thank you, Shreya. Once again, thank you.
Thank you, sir.
Thank you so much. So our next presenter is Miss Kalyani Ulas Deshmukh, also a PG student from Bharati Vidyapeeth College of Pharmacy, Navi Mumbai. She will be presenting on the topic of ensuring drug safety beyond trials, the promise of real-world evidence Her valuable researchers is guided by Dr. Vinita Vikhalvinkar. Please join me in welcoming Miss Kalyani Ulas Deshmukh. Miss Kalyani, you can join now. Please share your screen.
Yes, sir.
Bhavesh, I think she got left.
Sir.
Okay, she is back. Kalyani, can you start?
Kalyani, you can start and sharing the screen.
Yes, sir. I'm sharing screen. Thank you. Okay.
Kalyani, share your screen.
Yes, sir. I'm sure. Sir, can you give me two minutes? I'm unable to share my screen.
Bhavesh, you will take the next.
Okay. Our next presenter is Dr. Shashikala Metri, faculty at Gokhuraju Gankaraju College of Pharmacy, Hyderabad. She will be presenting her research on formulation and evolution of Polyherb ethosomal gel. Let's all welcome Dr. Shashikala Metri with a great enthusiasm.
Thank you, sir. Am I audible?
Yes, ma'am.
My screen is visible?
Yes.
So presentation also visible. Thank you. So today's presentation includes the formulation and evaluation of polyherbal ethosomal gel. So this is actually my deformed students book. They are not available. I will going to present it. Formulation and evaluation of this polyherbal gel, mainly it includes the polyherbs like clean herbs with 10 plus moringa contaminances and curcuma longa, which are already proven their anti-inflammatory activity through animal study as well as they are already there in the market. So we thought of going for the liposomal formation so that it will give efficient transdermal delivery of these active constituents present in these skin. So ethosomals, as already we know, these are the novel drug delivery systems which are very good for the transdermal delivery. So we formulated these three herbs in the ethosome suspension and then incorporated in the gel. What are the methods which we have used here? We have prepared the first, we prepared the extracts of these three herbs by maceration process, taking 100 gram of each of the drug and keeping it for 3 days and then filter. The filtrate was used for the preparation of the ethosomes. So how we prepare the ethosomes? The procedure mainly includes first we dissolve the specified amount of the lecithin and cholesterol in the ethanol. Then we have taken our specific weight of our drug and then we have incorporated in the ethosomes. Then the water is heated at the temperature of 30 to 40 degrees. The heated water is added to this mixture of the lecithin, cholesterol, and containing the drug. Drop wise at the speed of 700 to 1000 RPM using the magnetic stirrer. Then it was showing the appearance of the milky the opalescence. There how we confirm the formation of the ethosomes. Then those formulated ethosomes were evaluated by using a microscopic examination, by using a DLS, zeta potential, even the efficient entrapment capacity also we have studied for these ethosomes. So in this preparation of ethosomes, we have formulated in three categories. We use varying the concentration of the lecithin and the cholesterol and ethanol. So that I will discuss in the results. And after that, here we have selected one of the suspension. We have prepared three. Among that, the ethosomal 2 has shows the ideal properties, and we selected that for the formulation of polyherbal ethosomal gel. So here, for preparation of the gel, we have kept the carbonyl 940 dissolved in the water and left it for 24 hours, so that it will give a nice swelling and prepare the good gel. So then the gel was prepared in two phases. The methyl paraben and propyl paraben. Sorry to mention here, I have not mentioned the propyl paraben. We have used the propyl paraben also as a preservative. Methyl and propyl paraben both are dissolved in the water and added to this gel by stirring continuously. And then in the second phase, we have taken the ethosomal solution 3 ml, and this thing we have mixed it. Even the propylene glycol, I forgot to mention here, glycerin as well as ethosomal gel 3 ml, we have taken and mixed it and added to the carbopol gel with continuous stirring. So that complete mixture is then subjected for the pH adjustment using the triphenol amine. Then the gel was evaluated for its pH, viscosity, stability, readability, extrability, massability, and the patch test. And later we have subjected it for the in vitro anti-inflammatory activity. The results indicates is it visible, sir?
Yes, ma'am.
So results, phytochemical screening was carried out as a part of confirmation of the phytofunctions present in this. By using different chemical tests for the identification of alkaloids, flavonoids, phenols, amines, and saponins. All the three drugs shows the presence of alkaloids, flavonoids, and phenols, amines, then saponin, glycosides, and the sterols. The parijata flowers which we used shows the rich in the phenols and the flavonoids, as it has an orange stick, orange stalk. It is having more flavonoids and phenols in it. So in the evaluation of the ethosomes, as I have mentioned, that we have formulated three ethosomal suspensions, E1, E2, and E3, varying the concentration of the lecithin, cholesterol, and then the ethanol. So among these, the E2 shows the ideal properties of the ethosomes. So these are all the ideal properties like particle size, PDI, that is polydispersion index, zeta potential, and entrapment capacity also. It was good in the E2. So in case of E3, which we can see from the figures, even there is no formation of the proper shaped vesicles, whereas in case of E2, we can see the formation of E1 and also they are dispersed densely in the complete picture. And here you can see in the E3, there is no dispersion of the drugs, even means though they are means they are between the aggregates of the vesicles. So here we can, from these parameters, we have selected the E2 is the good ethosomes. Then that one we used for the formation of the gel. So here the gel which was formed by using the carbopol, propylene, glycerine, as already mentioned in the letter, the color of the gel was found to be yellowish with a rather pleasant. It was very smooth, and pH was found to be 5.8. It was good, and it is suitable for the skin conditions as it is we are using it for the anti-inflammatory activity, right? And anti-inflammation, and the consistency also found to be good. Viscosity was found to be 560. Spreadability, it shows very good spreadability around 14.5 gram per second. Then washability, it was easily washable. You can see there is no irritation observed when we applied on the skin. And extrability, easily it can easily come out from the tube. It was stable. Consider the stability. We have kept it for a month. We stored it for a month. There is no changes in the consistency. There is no growth of any microorganisms in this. So it was found to be stable. Patch test we did by applying under the elbow. So there is no allergic reaction also found. So here the same gel we have carried out the in vitro anti-inflammatory activity by using the protein denaturation assay, where we have prepared the different concentration of the gel as well as the standard. The standard used was a diclofenac sodium. Here it was found to be the in vitro concentration of the gel was found to be 47.87, whereas the diclofenac standard MSID shows 30.4. So we can, from this we can infer that our gel have the comparable result as that of the synthetic one, that is diclofenac sodium. So from this we conclude that the study demonstrated the formulation and evaluation of the polyherbal ethosomal gel incorporating the ethanolic extract of all these three drugs, which are well-known anti-inflammatory drugs. So anti-inflammatory formulation is substantiated again by in vitro analysis using the egg albumin denaturation assay. And future work includes the in vivo study of these drugs and also to check the synergistic action of these three drugs present in the formulation. So these are the references. Thank you for giving me the opportunity to present.
Thank you for the presentation, Dr. Shashikala.
Thank you.
Nice work. Just a couple of questions from my side.
Yes, sir. Yes, sir.
Hello, can you hear me?
Yeah, yes, sir.
Yeah, the analytical methods. Actually, have you carried out any analytical methods on the extract? And when you actually put. Yeah.
Analytical method, actually, it is the second part of our project, sir. In the first part, we have individually done the analysis of these drugs. So directly we have taken the results from that and we went for the interview study.
So just the reason of the question is because when we use the extract,
yes, sir.
There is most of the time there is a variation in the batch variation
Yes,
is there when you do the extraction.
Yes, sir. We also face the same problem. Actually, I have included 3 drugs here. So 3 drugs they have to show the same absorbance at the same nanometer. So here curcumin is yellow in color. Nictanthus subarctorpus also is having the very nice orange color because we used flowers here. Whereas Moringa concatensis. So all these 3 drugs individually we have taken the lambda nodes and we have kept the average of that lambda nodes. Always showing in between 400 to 500. So here Moringa concatensis showed the absorbance at 4 means nanometer absorbance at lambda nodes at 430. Whereas Nictanthus shows in between 444, Curculum Longa shows at 423-22. So we have taken the average of that and we checked the entrapment capacity of this equations. It is very difficult, otherwise we have to go for individually, so that is also can be done. But anyhow, these we are showing the 400 to 500, we kept at 430 as nanometer for the determination of amount of the drug.
Okay. And what about the any storage condition requirement for the gel?
No need, sir. It is stable. Even now also I am storing. It is almost 4 months over. There is no harm. Our consistency is same only. We can means full place should be there. There is not for specific conditions for that.
Okay. Thank you. Thank you.
Thanks.
Ma'am, so couple of questions from the audience.
Yes,
So first question is for how do the physiochemical properties of ethosomes influence the performance of the polyherbal ethosomal gels?
Sir, can you repeat again?
How do the physiochemical properties of ethosomes influence the performance of the polyherbal ethosomal gels?
Yeah, physicochemical properties like particle size, PDI, all these were displayed very nicely by this E2. So we prepared 3 ethosomal suspensions. Among that, the E2 shows a very good characteristic which are comparable to the ideal properties. So from that, we can confirm that we can include these, means we can select from this for the formulation of the gel.
Okay, the ma'am, next question is what tests are performed to check the stability of the polyherbal ethosomal gel over a time?
So 1 minute, sir. Once again, can you repeat it?
What tests are performed to check the stability of the polyherbal ethosome gels over a time?
Yes, sir. We could not get it. Actually, we could not get time to attend the stability test at high temperature, low temperature. Usually, for the herbals, these drastic conditions are not used. So, the normal temperature only we can keep it for longer time, for 1 month, 2 months, 3 months. In that way, we can go for the stability test. As these consist of the herbals, so we cannot go for the drastic conditions at high temperature. For synthetic drugs, we will do at high temperature, at very low temperature, at mid temperature. Such a stability test we cannot do for the formulation. So what we did, we have kept it for a month. So now also it is almost 4 months our project is completed. Even though it is not showing any, there is no growth of the microorganisms, there is no changes because they have just stored in the room temperature only. That only stability test we have done.
Ma'am, the last question is how does the EXVO skin permeability profile of the polyherbal ethosomal gel compare with that of the conventional polyherbal gel?
Sorry, yes, sir, we could not do this permeability test because of less time for the work. This is actually deeper work, so we couldn't get time for that. So in the next project, we are going to add all these things. Any more questions, sir? We couldn't do the permeability test.
Thank you, ma'am.
The drug release we didn't do, sir. Okay. Shall I stop sharing, sir?
Yes, ma'am. Miss Kalyani Deshmukh, you are able to join, share your screen.
Yes, sir. It has been visible.
Yeah, okay, you can start now, Kalyani.
Sorry for inconvenience.
Yes,
Good
okay, you can start
afternoon.
now.
My name is Kalyani. I am from. Okay, sir. Thank you. Good afternoon, everyone. My name is Kalyani. I am from Bharati Vidyapeeth College of Pharmacy, Belapur, Mumbai. And today's topic for presentation is the pharmacovigilance and real-world evidence. And the title for my poster is the pharmacovigilance beyond trials: the promise of real-world evidence. So there is always one question: are all drugs safe? We often assume they are, but the reality is complex. Since in 1960s there was a thalidomide tragedy. This tragedy showed us even seemingly safe drugs can have major consequences. The thalidomide was marketed as a safe and effective treatment for morning sickness, but later it leads to severe birth defects in thousands of children from this tragedy, and there's similar tragedies. The pharmacovigilance has come along the way. So the pharmacovigilance is simply the detection, assessment, understanding, and prevention of safety-related issues of marketed medications. The real-world data and real-world evidence are the two pillars of pharmacovigilance, and the integration of real-world evidence has emerged as a game changer in the field of pharmacovigilance. It helps to bridge the gap between clinical trials and everyday patient care. From analyzing data. So there are various sources of real-world evidence, such as social media platforms, spontaneous reporting systems, etc. The social media platforms increasingly recognized as valuable sources of real-world data, and currently, USFD and EMA acknowledge the social media potential to contribute real-world evidences, and it is based to use as a supplement data with the other sources. Next source is the spontaneous reporting system. It is the passive surveillance method where healthcare professionals, patients, and consumers voluntarily report the suspected adverse drug reactions to the regulatory authorities, and it is without the being promoted by formal studies. In India, there is a PVPI system, that is Pharmacovigilance Program of India, as a spontaneous reporting system, while in UK there is a yellow card system for adverse drug reporting, and in US FDA there is a MedWatch system. Another source is the electronic health records. These are the detailed longitudinal patient, including medical history, prescriptions, laboratory results, and clinical notes. Fourth one is the insurance claim database. Actually, it is the cost-effective way to collect the real-world evidences, because the insurance health insurance companies already collected the data, and we have to only analyze the data. Next source is the patient registry. It is the systematically gathers real-world data on population for a specific disease or specific condition with the goal of evaluating specific outcome over the time. Next source is the wearable and digital devices. For example, fitness trackers and digital watches. And also there is one more example, mobile health apps. There is one significance of these digital wearable devices. We can early detect the events such as heart attack and arrhythmia. So these are the significances. So these are the sources of the real world evidences. Now there are various significance of real world evidence by integrating real world evidences with the pharmacovigilance. The integration of real world evidence in pharmacovigilance has revolutionized the way we monitor the drug safety and effectiveness. And the real world evidences provide the valuable insight to drug safety and effectiveness. These insights, first one is a broader patient coverage. In case of clinical trials, there is a small population, and trials only done on a small population. But in case of real-world evidences or pharmacovigilance, there is a large population, and get the experiences. Next significance is the detection of rare adverse events. We can detect these real adverse effects which are unable to detect while the clinical trials. Actually, it uses real world experiences and real world evidence to make it easy to decision making for the regular authorities because of because there is real data is drug repurposing. It is based on significance. The term drug repurposing is therapeutic which are not previously known. So there is a huge data of pharmacovigilance to manage this data. There are such as the safety software, Viva World Safety Software, data software, with safety and pharmacovigilance software. Now let's explore the key regulatory considerations for integrating these Hello. Hello.
Yes,
Melanie,
Nalini.
you can share
Am I audible?
your screen.
Yes, sir. Is my screen visible?
Yeah,
Hello.
it is visible.
Okay, thank you, sir. So
Yeah, Kalyani,
let's yes, sir.
visible.
Okay, okay. So let's explore the key regulatory consideration. There are various regulatory authorities such as ICA, US FDA, European Medicine Agency, WHO, etc. So in ICS, the guideline ICH E2E and E2B R3 are the for the management of pharmacovigilance data. First one is the ICH E2E. This guideline is for the pharmacovigilance planning. It provides the structured framework for planning pharmacovigilance activities. And second is the ICH E2B R3 is a standard for the electronic transmission of individual case safety reports. Now, second regulatory authority is US FDA. It is governed by Title 21 CFR Part 314, which covers the post-marketing safety reporting and periodic adverse drug experiences reports. It regulates through the Act, US Food and Drug Cosmetic Act. Uh, in Europe, there is a European Medicine Agency, that is EMA, and good pharmacovigilance practices guidelines define standards for safety, signal detection, and risk management. Next is the WHO, that is World Health Organization, guides the member countries in developing pharmacovigilance systems, and it coordinates the reporting via VG-based database system. Now, the next part is the challenges for real-world evidence. Despite of the regulatory incorporation of real-world evidences into vigilance and its large data sources, there are still challenges to integrate the real-world evidences with the pharmacovigilance. Challenges such as signal detection delays. Actually, there are various causes of signal detection, such as poor quality data, incompleteness of data. There are some causes of da data for the signal detection delays. Next one challenge is the regulatory complexity. Uh, regulatory complexity. In regulatory complexity, there are various authorities such as FDA, EMA, PMDA of Japan have their own reporting standards. This needs company to navigate through varying obligations and timelines or frames, makes the regulation complex to the reporting. Next is the data integration. There is a
Now
large
you have
volume
2 minutes
of data.
left.
Hello.
Continue. Now you have 2 minutes left. Continue.
Yes, yes, sir. Due to large volume of data and different formats and standards, make it difficult to integrate real world evidences. So these are the challenges for the real world evidences. Now let's explore the what opportunities and future aspects can be done in the pharmacovigilance field. The AI, artificial intelligence and machine learning in pharmacovigilance can automate the processing of individual case safety reports and signal detection, and can duplicate the case identification and also reduce the manual working load on the working professional in the field of pharmacovigilance. It can predict, it can do the predictive analytics also, and by utilizing real world data sources and adding innovative real world sources in the field of pharmacovigilance, we can change the current situation into more insights and easy processing. And by global collaboration of various systems, we can harmonize the data of pharmacovigilance and overcome the challenges like heterogeneity of data. So this is all about pharmacovigilance and real world evidences. The integration of real-world evidence into pharmacovigilance ensures safe and more effective therapies. And we can say that ensuring drug safety beyond trial is the promise of real-world evidences. Thank you, sir.
Thank you so much, Kalyani, for your highly relevant and well-structured presentation. Now we invite our judge for questions and answer section.
Okay, sir. Thank you.
Thank you, Kalyani.
Thank you,
Can
sir.
you give an example where the social media platform was used in pharmacovigilance?
Yes, sir. News channels, even in the LinkedIn post. LinkedIn post, there is also the adverse drug related news we can see in LinkedIn. So they can uh, they can as a indication of adverse drug reaction in the specific area or specific region, and we can study on later.
Yeah, but how to collect that information?
Yes, actually the social media itself not act as source, but it can act as supplementary source to source with the other data sources. And yes, sir, by LinkedIn post or other social other news channels, we can get idea, and after that we can study these adverse reactions.
How is AI used in PV?
Actually, AI, artificial intelligence and machine learning, uh, it can duplicate. There is a data is available, and AI can analyze that data and classify it and use the results. And it can also use the analytics of data, predictive analysis of data.
It cannot be used to detect the signals?
Yes, sir, we can.
How? How?
From the previous information, if the previous data is available and the new consequences are happened, then the artificial intelligence can analyze the previous data and detect the signals as per their categories.
Okay, thank you. Thank you, Kalyan.
Kalyani, some questions from the viewers. First question is, what methodologies ensure the reliability and validity of real-world evidence in drug safety assessment?
Methodologies. There is regulatory authorities such as ICH, USFD, and EMA. We have to submit the data to these regulatory authorities. They use methodology. Actually, I sorry to say, but I don't know any methodology. But the regulatory authorities see and evaluate their data, and after that, we can ensure that the data is reliable and accurate.
So the next question is, what are the potential opportunities for enhancing patient reporting system in PV?
Yes, sir, we can promote the patients in the hospitals, and we can also promote the healthcare professional to report the adverse drug effect. As I am already say that the digital and electronic health records, patients can maintain easily the electronic health record or by using wearable digital devices.
Okay. Okay. Thank you so much, Kalyani.
Thank you, sir. Sorry for inconvenience.
Yeah, it's okay. Thank you. Thank you so much.
Thank you.
Now we will conclude the session, and the first representation of the session. His presentation was filled with innovative ideas, practical application, and remarkable research potentials. Heartfelt thank you to all our presenters and judges and attended audience for your participations. Now we will take a short lunch break, and we will back on 1:30 p.m. now. Thank you. Thank you so much.
Welcome back after the lunch, everyone. I hope you all had a refreshing and enjoyable break. Now let's continue the poster presentation session. The next set of presenter brings to us exciting research contribution in the field of nanomedicine, artificial intelligence, and nanoprotection. Let us begin with our first presenter after the lunch. I would like to call Ms. Vanitha M., a PG student from JSS College of Pharmacy, Mysore, Karnataka. Her topic name is Artificial Intelligence and Machine Learning Approaches for Early Diagnosis and Patient Satisfaction in Neurodegenerative Disorders. Her work is guided by Dr. Meghna GS. Let us call Ms. Vanitha to share her innovative research.
Good afternoon, ma'am. Thank you for the introduction. Just
Good
give me 2
afternoon.
seconds, ma'am. I'll
Okay.
share the screen.
Yes.
Is the screen is visible, ma'am?
Yes, ma'am.
Yes, ma'am. Shall I start, ma'am?
Yes, you can start.
Okay. Thank you, ma'am. Good afternoon, everyone. I'm Vanitha from JSS College of Pharmacy, Mysuru. And today, the topic for my poster presentation is Artificial Intelligence and Machine Learning Approaches for the Early Diagnosis and the Patient Stratification in Neurodegenerative Disorders. Coming to the introduction part, neurodegenerative disorders such as Alzheimer's, Parkinson, Huntington's, and amyotrophic lateral sclerosis are progressive and complex, with delayed detection often arising from overlapping symptoms and the lack of reliable early biomarkers. Advances in AI and ML are reshaping diagnostics by enabling earlier recognition of subtle disease signatures and accuracy through sophisticated pattern analysis and also by improving the patient stratification with integrated multimodal data. These innovations not only strengthen the clinical decision making, but also accelerate the therapeutic development and optimize the design of clinical trials, marking a transformative step in neurodegenerative research and the care. The main objective of the poster presentation is to evaluate AI and ML methodologies for the early diagnosis and patient stratification in the neurodegenerative disorders. The second objective is to highlight key techniques that includes CNN, SVM, random forest models, PCA that are applied in disease detection and progression modeling. What are all these abbreviations we'll be seeing in the discussion part. And the third objective is to analyze the current challenges, ethical and regulatory considerations, and outline future opportunities for the clinical integration. Methodology-wise, I have carried out groundwork through which I have identified challenges in orphan drug development for neurodegenerative disorder and the role of AI and ML in addressing them. Step 2, research phase I have carried out by analyzing the AI applications in early diagnosis, treatment monitoring, adaptive clinical trials, and the regulatory decisions for neurodegenerative orphan drugs. Step 3, I have compiled the structured findings into sections emphasizing technological benefits, regulatory challenges, and opportunities for AI integration in rare disease drug development. Coming to the discussion part, Machine learning is classified as supervised learning and unsupervised learning. First, we'll see what is supervised learning. Supervised learning is again subdivided as classification and regression. Under classification, support vector machine, that is SVM. This is classified. This is used for classifying the brain images. Example, MRI that tells the humor detection. Second one is naive bias that for classifying early Alzheimer's disease risk from MRI and PET data. Third one is convolutional neural network CNNS for the structural imaging that includes MRI, PET, CT scans, etc. Second one is regression. In regression, first one is linear regression that is used for predicting the cognitive decline scores from MRI volumes. Next one is ensembles. This mainly focuses on the gradient boosting for predicting survival rates using MRI plus PET data. Third one is recurrent neural network RNN for sequential data that tells the series from EEG and MEG. Next classification is about unsupervised learning that talks about the clustering. Clustering mainly tells about the hierarchical clustering for PET brain activity patterns and hidden Markov models HMM for sequential data from EEG and MEG. Generative adversarial networks GAN for generating the MRI images for training the deep learning models that executively tells about the neurodegenerative stages in a patient's brain. Next, we'll see about the deep learning techniques that mainly focuses on image analysis and diagnosis with methods such as CNN, RNN, and GAN, which we have seen previously. And the process involved in the deep learning is it first undergoes the image segmentation, then feature extraction, then modeling and analysis of the image, and then the decision support will be given with proper evidence. Next is neuroimaging approaches. This mainly focuses on the brain structure and function monitoring with techniques such as MRI, PET, and CT scans that has both structural and functional imaging. Next is EEG and MEG. This mainly talks about the brain activity. NIRS totally tells about the blood flow and the oxygenation that is involved in the brain capacity. The process involved in neuroimaging approaches are signal acquisition and processing, feature extraction, and deep learning classification. Central integration combines algorithms and big data for analytical insights and enables the assistive devices via real-time processing and the feedback. The process again continues with the input images. These are raw images collected from the patients, often used to detect abnormalities like tumors, hemorrhage, or structural changes. Deep learning process with this input images are processed as CNNS, that is good at recognizing the spatial patterns like tumors or lesions. After CNN identifies the tumors, RNN comes into picture. It shows the suitable for analyzing time series data such as changes over time in MRI or EEG data. Next comes GAN into picture. This generates synthetic brain images and enhances them by filling gaps, improving resolution, or simulating the disease progression. The final output will be the processed data, which is classified as glioma, hemorrhage, stroke, or it is a normal kind of brain. Everything will be classified in the output data using this ML models and AI techniques. Coming to the result part, challenges wise, yes, ML has challenges too. What are all those means? It has data limitations, model interpretability, transparency, standardization and clinical adoption, and moreover, limited high quality data sets are lagging, and ethical concerns that are related to the data privacy and the bias is still existing, and the need for clear regulatory framework need to be developed. Ethical and legal considerations are bias in AI models that do brings risk of misdiagnosis due to biased training datasets, informed consent from the patients, those who are involved, ensuring transparency in AI-assisted decision-making, regulatory compliance, adherence to guidelines for AI in healthcare, future perspectives, development of larger and diverse data sets to improve the model training, advancement in federated learning to ensure data privacy while training AI models, enhancing AI clinician collaboration for improved diagnostic decision making. Conclusion wise, AI and ML facilitate early detection and precise diagnosis and the patient stratification in neurodegenerative disorders. The 2025 approval of ONAPGO drug exemplifies the therapeutic and regulatory convergence. This emphasizes AI's transformative potential and the imperative for collaborative, responsible integration in the neurology field. These advances collectively signal a new era in technology-driven care for neurodegenerative disorders. These are all my references, and thank you for this opportunity.
Thank you so much, Vanita, for that highly insightful presentation. Now, I would like to invite our judges and audience to raise the questions.
Hello. Thank you for the presentation.
Yes, sir.
Nita. So just got a couple of questions. So from where
Yes, sir.
the data was taken to develop the model?
So the previously available brain models like from affected patients, those CT scans and MRI reports were collected, and those data were incorporated, like given for reference for this ML models. And like those ML models, after taking this data, they'll collect the data and evidence from this previous history of patients, those who were affected. From them, this ML models will take data. Okay, this is my problem. Through this only the patient is affected, like that. They'll get a clear-cut idea. And AI techniques comes into picture. For example, ONAPGO, which is FDA approved in 2025. This is the recent AI tool for mainly related to neurology. This AI tool collects the data from ML models and it gives us the solution. This can be the reason for the loss of neurons. Like that, a proper analysis will be made and the final output comes, sir. So that it will be helpful for the neurologist to quickly track the reasons for the neurodegenerative disorders in patients.
Yeah, but when you are saying that you took the data which is already there,
yes,
were there any non-disease images?
No, sir. These were proven disorders, sir. Like those were already affected, and neurodegenerative disorder is already a rare kind of disorders. In US and all, they define neurodegenerative disorder as rare because it affects only 1 or 2 in 5 lakh population. So it is such a rare disorder. So since it is rare, the patients, those who were affected, where their scan reports and their medication reports were tracked and kept for future references. From those data only, these ML models are given the evidences, sir. So it is the chances of fake results are less only, sir.
But how can you compare them? Because you need to have disease and non-disease images to train the machine.
Yes, sir. Disease condition, they'll be the ML models will be taken from the past history and all. Non-disease, a normal human brain's CT scan will be compared, sir, through clinical trials. A normal human being and the past histories, human brain CT scans will be compared, sir. MRI scans, that and all.
So how much data was used?
I'm not sure about it, sir. Since the development is in clinical trial stage 3, sir. So as of now, only few data are used, sir.
So when you say large database is needed, which how much database is needed?
I'm not sure about it, sir.
Because in the challenges you mentioned that
Yes,
high
yes, sir.
quality data sets are needed.
Yes, high quality data sets in the sense the data sets or data like it should be capable of avoiding data breaches, and mainly it should be concerned about the data privacy. In that aspects, high quality data sets are needed. So it is a main limitation like that I mentioned, sir.
Okay. Thank you.
Yes, sir. Yes, sir. Thank you, sir.
There are a couple of questions from our viewers.
Yes,
First
ma'am.
question, what roles does patient stratification play in the management of neurodegenerative disorders, and how can AI help in this process?
Yeah, generally, patient stratification term defines about right treatment for the right patient at right time. So this is achieved by a clinical trial process, which we all know. Clinical trial process is nothing but testing a new drug on the patients and the healthy volunteers to give a proper treatment for the patients. So, in this aspect, patient stratification in neurodegenerative disorders is achieved through AI and ML models. By ML models are given with the past history data which from the affected patients so that they analyze them, and AI tools such as ONAPCO and many other CNN, RNN, GANN. These are all the AI tools which are incorporated with the ML models input data, and through that the AI will suggest what are all the improvements that can be made or the changes that can be made in the brain signals to avoid the future recurrence of progression in the neurodegenerative disorder. So after incorporating all the results, obviously they cannot test directly on humans. For that also AI models are available in which those AI models have the practical brain how human has the same way. So through testing on them, we can lively see the progression in decrease in the neurodegenerative disorders as we take the drug. So drug is also given, and after that, how a brain is reacting, an affected brain is reacting to it. Like that, this AI tools will track, keep a track on it, and then it will deliver the output results so that we can compare and easily take out the result. Okay, this drug is more effective, and this is more effective, this is less effective. Like that we can analyze, and it mainly helps the clinicians to give a proper report to the doctors that this drug is very good enough. Like that it is more helpful.
Okay. Thank you, Vanida, for your wonderful presentation.
Yes, ma'am. Thank you, ma'am.
Yes, yes. Now our next presenter, Mrs. Sonal Ramdev Berde, a PhD scholar from Pune College of Pharmacy, Bharati Vidyapeeth Limited University. She will be presenting on in-silico approach and molecular docking of kaolin for neuroprotective study. Her research is guided by Dr. Deepa S. Manlik. Ma'am, can you start your presentation?
Yes, Saksha, I'm audible?
Yes, ma'am.
Okay, my screen is visible?
Yes, yes.
Okay. Good afternoon, one and all present in meeting. My presenting topic is in silico in vitro identification and molecular docking of kaolin for neuroprotective activity. The abstract presents the Alzheimer's disease is a progressive neurodegenerative disorder associated with aging with the progressive hallmarks. The amine isnega, a natural source for kaolin, has showing the traditional claim to use its role in Alzheimer's disease, to use in the various diseases like Parkinson, various other cardiovascular diseases, but it is remain in the Alzheimer's disease to showing the exploration. In this study, the Alzheimer's disease related GeneCards with compared with the kaolin targets by Swiss target prediction were analyzed through the network pharmacology, drug likeliness by the moles of pre-ADME parameters, then protein-protein interaction, cake enrichment, Go analysis, past prediction, and with the molecular docking. Among these, the Alzheimer's disease target genes and the kaolin targets were overlaps and identifying Then molecular docking showed the Khelin binding comparable to the donepezil, which suggesting the potential multi-target therapeutic role of Khelin in the Alzheimer's disease. Along with that, the various cytotoxicity assay like Brainstorm lethality assay and the Allium-SEPA mitotic index was also performed for the safety assessment identification of the Khelin. The material and method for the in silico as well as in vitro and molecular docking approach, the Alzheimer's disease related targets were retrieved from the GeneCards and the Khelin targets were predicted from the Swiss target prediction. Then the compound target disease network were built using the Cytoscape. Here, firstly the collection of Khelin from Amibisnega by searching the literature search of EON impact database, and the Khelin was selected based on the literature search on the PubChem. Here is the graphical abstract showing the Khelin literature search. After that, there is the collecting of the targets of Alzheimer's disease was collected through the ADME study in the PubChem. The Khelin showing the bioavailability, blood-brain barrier, the all parameter to be shown. The Khelin has to pass. That blood-brain barrier also showing as well as the high bioavailability. The past prediction were also conducted to showing the various mechanisms of Khelin showing the partially active, its amount and the partially inactive. Then molecular docking was carried out through the Schrodinger's Maestro glide, which evaluated the binding of AD targets. This molecular docking carried out by involving the three steps are there. That is the target prediction, target preparation, ligand preparation, target ligand docking here. The molecular docking was done using this glide in order to examine the chosen compound mechanism of action. Binding affinity, binding mode, and molecular interaction by the Maestro panel were utilized to construct the Khelin and donepezil networks was acquired to protein data bank and docking ready with showing the protein preparation wizard. And the binding affinity 2D and 3D structures were plotted. Then the Brainstorm lethality assay showing the particular LC50 values by comparing the showing two principle, it based on the ability of test substance to kill the Artemisia salina larvae, and the lethality of compound to larvae is indicative of its potential cytotoxic anti-tumor as well as pesticidal properties. So first step is the hatching of Brainstorm, preparation of test samples, then give the exposure to test substances, its incubation period, observation, and data recording, and the data analysis. The LC50 value, interpretation of the LC50 values indicates the toxicity level to showing it is safer or not, showing the safe line for the further study. Then antimitotic Allium-SEPA study was performed on the Allium-SEPA. Its principle, the principle of the Allium-SEPA assay is to determine the ability of test extract to interfere with the mitotic process in the root tip cells of the onion. The root meristematic cells have highly mitotic, and any disturbance in cell division can indicate the antimitotic and cytotoxic properties of the tested compound. This step, if we there is given the stepwise how these antimitotic assay carried out, the preparation of onion bulbs, then treatment with the extract given, then fixation and staining, microscopic observation, and the data analysis. The significant reduction in the mitotic index indicates the strong antimitotic potential, which suggesting the compound or extract possess the antiproliferative or the cytotoxic property. The result and the discussion showing the core target of a PPI network of drug in Alzheimer's disease. Firstly, through the Swiss ADME study, the bioavailability, the BBB passing was carried out. Then there is a core target of PPI network of drug as well as Kelin, and the AD found the STAT3, Cas3, HSP, then TLR4, RELA, PTGS2, mTOR, CXL. These are the various genes which are showing the betweenness and centrality and the drug centrality more as compared to the other. Apart from these genes are selected for the further molecular docking study, and the molecular result was docking result was found by the docking score. Here, these are the certain core target genes are there: Cas3, HSP90A, CXCL8, PTGS2, NF-kPIA, RELA, mTOR, MAPK14. These are the various genes. Compared molecular docking compared of Kelin with the donepezil. As if you have to see the figures here, the HSP90, PTGS, NF-kPIA, RELA, mTOR, and MAPK showing the binding affinity as compared to the donepezil. It showing the high binding affinity is there. Sorry. After that, if you have to focus on the past prediction results are there, through which the anaphylactic receptor antagonist, multiple sclerosis, HIF1A expression, CYP1A, inhibitor, apoptotic, antimutagenic. These are the certain pathways or mechanisms which are partially active by the Kelin mechanism to show. Apart from this,
Ma'am, you
the
have 2
Kelin.
minutes left now. Continue.
Hello.
You have only 2 minutes left now, ma'am. Continue.
Okay, yes, thank you. Apart from these brine shrimp as well as the mitotic assay showing the good activity and safer for the further use of the Kelin. These are the 2D and 3D structures of the Kelin comparison with compared with the donepezil. Apart from this, the discussion was the network pharmacology shows the protein binding network with the various targeted genes are there, that is the STAT3, Cas3. Amongst these, MAPK14, mTOR, RELA, these showing the very good binding affinity as compared to the donepezil. Then this BSLA screens toxicity, Kelin showing the brine shrimp lethality assay, which is safer for proceed to neuronal cell line study as neurodegenerative SHSY5Y. Kelin also inhibit the mitosis and causes the chromosomal aberrations, which is safe for the neurogenesis. The all study of in-silico in vitro assay as well as toxicity assay, it conclude the molecular docking study demonstrate that Kelin exhibit the comparable binding .
Thank you, ma'am, for the well-structured and valuable presentation. The questions from the judges.
Yes, I'm audible.
Yeah, you're audible.
Actually, lastला I think unmute झालं होतं.
Yeah, you can continue, ma'am, if there is anything left.
Yes, it is conclude that the toxicity assay demonstrate showing the low toxicity supporting it is safe for the cell line study. And the Allen-Sepa mitotic assay showing the Kelin did not cause any notable mitotic interference. Apart from this, the molecular docking also showing the good result of Kelin as compared to the donepezil, which showing the good binding affinity. These are all references, and thank you.
Thank you, madam, for the
Thank you.
presentation. There are a couple of questions. So the molecule which you are using, Kelin, it is poorly soluble in water.
Yes, sir.
So when you do the essay on the brine shrimp,
Yes,
how
sir.
was the sample prepared and what were the concentrations?
It is soluble in DMBA. So I have made the samples in DMBA.
DMBA. And what was the concentration?
Hello?
Hello.
Sir.
What was the concentration you used on the
For the
brine shrimp?
brine shrimp, 1 ml, 2 ml, likewise 3 ml, 4 ml. Up to the 5 ml, I have to make the samples are there through the stock solution. Then 50 microgram, 100 microgram, making the stock solution, and from that 1 ml, 2 ml, I have to take the stock from the stock.
Okay. And in the alum Cephas assay, where it is mostly utilized to understand antimitotic or chromosome aberration, what was the evolution criteria? What actually you looked in that assay?
The cell was live or dead, depending on that. The micro.
Okay. Thank you. Thank you.
Yes, thank you, sir.
There are a couple of questions from the audience also. First question, how do the chosen in silico models and docking protocols compare to other established methods, and what are the limitations of these computational approaches?
Sorry, I didn't get the question. First question, first.
How do the chosen in silico models and docking protocols compare to other established methods?
Yes.
And what are the limitations of the computational approaches?
Yes. For basic literature purpose, this network pharmacology or in silico approach of network pharmacology, it is the I can say that it is the base of study through which we can find out the major target which we want to incorporate in our study. So through this study, I can find the core target from the network PPI network, and from that I get the molecular docking target, and from that I dock the Kelin with donepezil. So this is the major role of in silico study.
Okay, thank you, ma'am, for your presentation.
What is the second question? Okay, thank you.
Now, to continue with our existing lineup of questions, we will hand over the next part of this session to Preeti Shirsagar. Preeti?
Yes, Sakshi, can I audible?
Yes, you are audible.
Okay. Good afternoon to everyone. On behalf of Swalife Foundation, I extend a warm welcome to all the faculty members, judges, and participants present here today. Poster presentation is not just about displaying information. It is about presenting the complex ideas in a clear, concise, and visual appealing way. It gives a students and a researchers a platform to share their work, interact, and with experts and enhance their knowledge. So, without further delay, let's begin with the presentations. I invite our next participant, Vaibhav Jain, from the Research Scholar, Department of Pharmaceutical Analysis, Parole Institute of Pharmacy. to present their poster on a GAPI, an assessment tool of green chemistry. Vaibhav,
Thank you, ma'am, for the introduction. Yes, am I audible to you?
Yes, or, you are audible.
Okay, ma'am.
You can share your presentation.
Yes, ma'am. Ma'am, is my screen visible?
Yes, screen is visible.
Okay, ma'am. Thank you. So, a very good afternoon to one and all present here. As introduced by ma'am, my topic for today is GAPI, an assessment tool of green chemistry. So, we shall start with what is green chemistry. Green chemistry is an emergent domain. Green chemistry is an emerging domain in chemistry that especially emphasizes those approaches that avoid the involvement of harmful substances, products, or we can say the chemicals that can cause serious harm to any individual or to the environment. This concept is solely based on the principles introduced. In case of analytical chemistry, green chemistry promotes the use of environmentally safe and less toxic chemicals and instruments. For this need of an assessment tool that determines whether an analytical method is complying with the concept of green chemistry or not. The basic concept of green chemistry is based on its 12 principles. The principles involve pollution prevention, atom economy, less hazardous chemical synthesis, designing of safer chemicals and safer solvents and auxiliaries, design of energy efficiency, use of renewable feedstocks, reduce derivatives, catalysis, design for degradation, real-time analysis of pollution prevention, and inherently safer chemistry for prevention. If we look into the GAPI, then GAPI or the Green Analytical Procedure Index is an assessment tool was developed by a Polish scientist named J. Plotka Veselka in 2018. This concept is solely based on the green principles, as I explained earlier. This provides a numerical value for method which range between 0 to 100. The closer the value the closer the value towards the 100 is considered, the more greener the method. It is generally used for determining the greenness of the analytical method by considering various aspects, as we can see in the GAPI pictogram. This tool mainly quantifies the greenness of any method based on sample, preparation, reagents, and compounds used, along with instrumentation, general method type, and sample collection, prevention, transport, and storage. We can see the GAPI pictogram. This is how it gives its numbering and analyzes the various aspects of any analytical method. Now, if we look for the pros and cons, it has very comprehensive coverage. It helps identify the weak stages where uh, where it needs to be improved. Provide a numerical store that determines that the compatibility of the analytical method with the green principles. If we take about the disadvantages or the cons of this tool, steps are difficult to understand somehow to any individual who is using or who is not very comfortable with the use of this tool. Modified versions such as AgriPrep, etc., are now available. We can see them as in recent developments as well. Agri, RGB12, and AgriPrep. These tools also used to assess the greenness of any analytical method the similar by considering the similar principles of green chemistry as done by GAPI, but it gives more precise and more accurate values as compared to this. GAPI, if I conclude, GAPI or a green analytical procedure index is a metric tool, the system that assesses the greenness of any analytical method by evaluating various aspects of any technique, including sample preparation, reagents, compounds used, instrumentation, sample collection, preservation, and transport and storage. This comes after the need for developing a metric system that determines a numerical value for determining the greenness of any method, or we can say that analytical method. This gives the numerical value ranging from 0 to 100. The higher the value, the greener the method is considered. In recent years, many other developments for the metric systems have been introduced. As I said earlier, in the recent developments block. So this is how I would like to conclude. We can see the references from where I have taken. Thank you.
Thank you, Vaibhav, for your wonderful presentation. Now I
Yes.
request our judges and audience if they have any questions to kindly ask the presenter.
Thank you, Vaibhav, for the presentation.
Yes.
So how can we actually use the GAPI in the drug discovery?
So in case of drug discovery, we can consider the greener method for discovering a drug, or we can establish a more greener method. Like if we involve, in case of if we talk about the analytical aspects of the discovery, we can consider more green solvents like ethanol and other solvents. Answer for enhancing solubility, there is a term in pharmaceutics that is hydrotrophy or hydrotropism, which involves salts of certain compounds that like sodium acetate, sodium benzoate, that when mixed in stronger concentrations, it can enhance the solubility of a drug. So that's how we can it can play a vital role in drug development.
Okay, so recently there is a development in the GAPI. There is a modified GAPI tool
Yes,
has
sir,
been.
modified.
Yeah. So what is the difference between that?
So modified GAPI inspects more more aspects. Like we can see in the GAPI pictogram, there are sample preparation, sample collection, preservation. Similarly, sir, it gives more precise value as compared. However, sir, I have just started reading about these matrices, so I have not reached up to that level as I am still reading about that.
Thank you. Thank you.
Yes.
Vaibhav, some questions from the audience.
Yes, ma'am.
Your first question, how does GAPI help in evaluating whether an analytical method is environmental friendly?
Ma'am, it actually assesses whether, like, in case of HPLC method development, we can say use of ACN or acetonitrile is far more toxic if we compare the greener solvents like methanol and ethanol. So, if we are discarding these solvents after HPLC a total run, it will still cause an harm to the environment if we are discarding it without preparation. Right. So it directly contradicts with the green principles like atom economy and less hazardous chemical synthesis designed for degradation and use of renewable stocks. Right. So in that way, we can say that uh, this is how it plays a vital role in considering it can shows that use of which solvent can develop a more greener method in HPLC. So this is how it can give.
Okay. Your second question is, what is the purpose of assessment tool in green chemistry?
Ma'am, the principles of green chemistry, as I've shown here, ma'am, these principles are need to be evaluated over time. Like after development of a method, the principles are met or not. Like we validate a method after developing any analytical method. We validate a method. Similarly, we have to validate whether the various aspects of any analytical methods are meeting with the green principles or not. This is why we need the matrices or analytical tools to assess that whether a developed analytical method are complying with the principles of green chemistry or not.
Okay. Next question is, what are the limitations or challenges of using standardized assessment tools for green chemistry in industrial applications?
Sorry, ma'am, can you please repeat your question?
Yeah. What are the limitations or challenges of using standardized assessment tools for green chemistry in industrial applications?
Ma'am, in case of industrial aspects, ma'am, we can say that at industrial level, we can say that however in many cases, not in some cases, there are nearly impossible to decrease the waste, as when we produce something or when we discover something, we have to do trial and error method, or we have to make a big batch in case of production of any drug. So ma'am, we can consider that it has many challenges, but ma'am, it has possible results as well.
Okay. Thank you, Vaibhav.
Yes, thank you.
Now, I invite our next participant, Vivek Kode from Pearl Institute of Pharmacy and Research, to present their poster on hydrotropes and a green solution for a sustainable future.
Good afternoon, sir, and good afternoon, ma'am. Hello, ma'am.
Yes.
My screen is visible.
No. Your screen is not visible.
Now it is visible, ma'am.
No.
Actually, there is a network problem, ma'am.
You can try again, Viveka.
Yes, ma'am.
Viveka, you can try to share your screen.
Sir, I tried, but it is not. Sir, हो ही नहीं रहा है. So now it is visible.
Yes, Viveka, your screen is visible now.
Thank you, ma'am. I am Viveka Kode and I am from Parul University. And my topic is hydrotropes, a green solution for a sustainable future. First of all, what is hydrotropes? The hydrotropes are a class of compounds that increase the solubility of hydrophobic substance in aqueous solutions like water and all. Unlike surfactant, they do not have a large enough hydrophobic part to form micelles spontaneously. Instead, they enhance solubility by interacting with the solute and the water molecules, helping to couple the two together. There is a mechanism. First is self-aggregation, then it's form micelles, and then structure is disrupt and structure is creator. Then methods. There is a reported method, titrimetric, spectroscopic, and chromatographic. Here is a title for titrimetric, a novel and validated eco-friendly titrimetric method for the estimation of furosemide in a solid dosage form. There is a description. In this article, the use of hydrotropic concentration is a 5% urea solution. Then solubility enhancement rate is 10 folds. Then titrant is used, zero point 0.1 molar NaOH. Then indicator is used, bromothymol blue. Then second is a spectroscopic method, analytical method development and validation of spectroscopic estimation of ibuprofen using hydrotropic solubilization technique. Then there is a description. The hydrotropic concentration is used 2 molar ammonium acetate and 2 molar sodium benzoate. Then solubility enhancement rate 20 folds and the wavelength detection rate 264 nanometer. Then chromatographic method, title, application of an eco-friendly cost-effective hydrotropic solution as mobile phase for the estimation and validation of onidazole in bulk and pharmaceutical formulation by RPHPLC. Then use of stationary phase is Shimadzu Shimpac C18. The column size is 4.6 into 250 mm and 5 micrometer particle size. A mobile phase is used, 5% urea of solution. Then flow rate is 1 ml per minute. Injection volume is 20 microliter. Then retention time is 3.996 minutes. The detection wavelength 320 nanometer. then it is we can apply this in a green analytical chemistry. The green analytical chemistry is an approach that aims to make analytical processes more environment friendly and safer for human health. It extends the 12 principle of green chemistry to the specific context of chemical analysis. There is a 12 principle behind green analytical chemistry. The first one is pollution prevention, atom economic, less hazardous chemical synthesis, design safer chemicals, reduce derivatives, use of renewable feedstocks, design for energy efficiency, safer solvent and auxiliaries, catalysis, design for degradation, real-time analysis for pollution prevention. Then last one is inherently safer chemistry for accident prevention. There is a greenness assessment tool. We can assess the how our method is green and what is the percentage of it. Uh, these are quantitative and qualitative framework used to evaluate and compare the environmental and health impact of different analytical methods. They help chemists choose the greenness procedure. There is a two greenness tools. First is AGRI and second is CAPI. The AGRI, it means the full form of AGRI is analytical greenness metric. This is compressively software based tool that assesses an analytical. It generates an easy to read circular pictogram with 12 segments. Each segment represent one principle and is colored on a red, yellow, green scale to indicate its greenness score from least to most green. The overall score is displayed in the center. Then GAPI. GAPI full form is a Green Analytical Procedure Index. This metric uses a pictogram with 5 pentagrams to evaluate the greenness of a procedure from the start to finish. It covers different stages from a sample collection to final analysis and visually represent the environment friendly of each steps using a red, yellow, green scale. Then the last is conclusion. Hydrotropia are the best method to improve the aqueous solubility of low and sparingly water soluble drugs. These techniques are non-toxic, straightforward or simple, and green methods have been developed because of their eco-friendliness. These aim to provide information on the handling and selectivity of suitable criteria for analytical methods, that is titrimetric, spectroscopic, and chromatographic methods by using hydrotropic solubilization. Thank you. And there is a reference.
Thank you, Viveka, for your excellent presentation. Audience and judges can ask the question to the presenter.
Hello.
Yes, sir.
Yeah, thank you. Thank you for the presentation, Viveka.
Yes, sir.
So just the question regarding hydrotropes. Can you
Yes.
give me the example of a hydrotropes
Yes,
which
sir.
can be useful in a in the pharmaceutical formulation development?
Sir, urea, niacinamide, sodium benzoate, sodium citrate, sodium benzoate, sodium salicylates. Sir, these all are the examples of hydrotropes which enhance the solubility.
Okay, and what happen if they are combined with the other molecule? Do they interact with the other compound?
Yes, sir. It interact with compounds to enhance the solubility of compounds. If compounds is non-soluble, it enhance the solubility of that compound.
Does it change the role of that compound?
No sir, no changes in compound like pH हो गया और भी बाकी के जो भी पैरामीटर्स हो गए उसमें कोई चेंजेस नहीं आएंगे, और आ भी गए तो सर इसका कोई ऐसे इफेक्ट नहीं होगा, नेगेटिव इफेक्ट नहीं होगा सर.
So then why do we need other methods? Like why we do nanoparticles? Why don't we just use the hydrotropes?
Sir, it's hydrotropes is a green related to the green chemistry. We use other also, but this is develop a green method because of it we use.
Yeah, that's what I'm saying. If they are so useful, why when we want to use a non-polar compound, instead of going for other technology, why don't we do
Sir,
hydrotropics?
sir, it is cheap. Sir, we use solvents, organic solvent in RPHPLC, sir. But that cost is high rather than a hydrotropes. It is too much cheap, sir.
Thank you.
Okay, sir. Thank you, sir.
Viveka, some questions from the audience. Your first question is, how does the use of an assessment tool in green chemistry facilitate the design of more sustainable chemical processes?
Sir, ma'am, please can you repeat?
How does the use of an assessment tool in green chemistry facilitate the design of more sustainable chemical processes?
Actually, ma'am, these two tools are used to check the greenness of our method, and
Next question is, what role do assessment tools play in improving the environmental and health safety of chemical products?
what, ma'am?
What role do assessment tools play in improving the environmental and health safety of chemical products?
No idea, ma'am.
Okay. Next question. Can you give example of natural or bio-based hydrotropes used today?
It's urea, niacinamides, sodium benzoate, sodium citrate.
Okay. Next question is, in what ways does a green chemistry assessment tool promote the development of safer chemical products?
Yeah, it is used for several products. Uh,
Okay, thank you, Viveka.
yes, ma'am. Thank you, ma'am.
Thank you. Next compelling hand over to Bade sir.
Thank you, Preeti. So all the poster presentations we have finished now, and our session, which we have will be starting at 3 p.m. I have already communicated with the speaker, and he will be joining early. So hopefully, if he join early, then we will start the session before 3 p.m.
Good afternoon, everyone. I'm Miss Safa Saudagar, and it's my pleasure to welcome you to this session. Today, we are honored to have with us Dr. Ahmad Zia Haidari, who will be conducting a session on temporal modeling of fibroblast responses to cancer, which are derived extracellular particles. Dr. Ahmad, he is a director of studies for PhD and professional doctorate. He is a senior lecturer and a course leader of MSc Molecular Biology and Health at the University of Stafford. So, without wasting much of time, I warmly welcome Dr. Ahmad to share his valuable knowledge with us.
Thank you very much. Thank you for providing this opportunity. And I'm sorry, I'm a bit late. Probably I was in a meeting. So you introduced me. The university is called the University of Staffordshire. It's in the Midlands in the UK. Just for the reference for those who are listening. Yes. So I would like to share my presentation with you. Are you able to see my screen? Just confirm.
Yes, yes.
Thank you very much. Does my voice is clear, Dr. Praveen?
Yeah, your voice is clear.
All right, thank you very much. Our talk is today on temporal modeling of fibroblast responses to cancer-derived extracellular particles crosstalk in the tumor microenvironment. Our discussion will be based on a brief introduction on the topic of cancer first. And then we move on to discuss the importance of extracellular particles, the biogenesis and the function, and their role in the tumor microenvironment. And then we discuss the extracellular matrix components of tumor microenvironment and their role, and the stroma cells, focusing on fibroblasts and the biology and their role in tumor genesis, and local invasion, and also the importance of crosstalk between cells in the tumor microenvironment. And then what's the implication of this crosstalk, and then how we can move on to apply this knowledge in the context of cancer genesis in terms of diagnosis, prognosis, treatment, and monitoring treatment. So we're starting off by a quote from World Health Organization, reported back in 2022. They said 20 million people were diagnosed by cancer in general, and this is worldwide, globally. So 20 million people were diagnosed, and 80 to 90% of cancer is derived from epithelial tissue. So if we consider 80%, then that means 16 million of people diagnosed with cancer of epithelial tissue. If it's 90%, then it would be 18 million people. So that means a very massive proportion of cancer is derived from epithelial cells, epithelial tissue. So one of the questions you may think of is, say, why epithelial tissue? The body is composed of 4 types of tissue. Why epithelial tissue is accounting for 80 to 90% of cancer. And they also estimated that by 2050, then this figure will increase to 35 million per year. So one of the questions never ever putting forward in any conference, if you guys attend, is we are scaring people for, this is the figure, millions of people diagnosed. But please ask this question from whoever putting this figure forward. 20 million, in this case, 20 million people are diagnosed. So I'm 100% sure they have the figure of how many millions of out of these 20 are diagnosed with what stage. Why we don't have this figure? It's important. We will discuss this, why it's important, right? It's important because 90% of patients die from cancer as a result of metastasis, right? If over 90% of people die from cancer. And then what's the percentage of survival of cancer? Let's say in 2022, if they're saying 20 million people diagnosed, so what's the survival rate? The survival rate is significantly low. Why? What's the question? What's the answer for this? So the answer for this question is the diagnostic procedure has got some problem. Yes, we managed to diagnose cancer, but at what stage? Is that the stage we are diagnosing the cancer can help the patient, can help the society? Here we're talking about the NHS, the government. How much money a single patient suffer from cancer is needed to diagnose, to confirm the diagnosis, that person definitely has the cancer. And then we move on to the next step. What type of treatment is available? And what's the survival rate? Spending millions of pounds, dollar or whatever currency, depends on the government. At the end of the day, what we are saying, we say sorry. It's at the late stage. It's a very late stage. The possibility, the survival rate is 6 months or a year. Why? Right. This is a question. We can discuss this. So this presentation is based on this question. So we have a massive problem in cancer diagnosis. Yes, treatment is good, of course. We need to focus on personalized medicine, but we need to focus on personalized diagnosis, right? If we don't fix the diagnostic procedure, we can't fix this problem. We can't fix this issue, right? If cancer is metastasized, you can't defeat it. We definitely lose. right? So let's move on to discuss then. So what's the problem then with this disease? Cancer is not just a disease of malignant cells. So what does that mean? So remember, we are focusing on carcinoma, which account for 80 to 90% of cancer, right? So you think about not cancer cells, then in this case, we're talking about cancer tissue, or let's say tumor tissue, right? So if you observe and if you analyze a tumor tissue, you can literally see less than 20% of tumor is composed of malignant cells. So vast majority of tumor tissue is composed of other type of cells, right? So what are they? Those cells called stromal cells, right? Stromal cells. Stromal cells such as fibroblast, such as macrophages, such as dendritic cells, such as immune cells, endothelial cells, and biological molecules, most importantly, such as collagen, elastin fibers. So these are the rest of the components of the tumor tissue. Now, we are focusing and we are targeting cancer cells. That's fine. We are trying to design anti-cancer drug to target cancer cells. But we need to pay attention on this 80% or 70% of the component of the tumor tissue. If you focus on these cells and if you focus on the biological molecules can be found within the tumor tissue, the first question is, are they normal? The fibroblast is normal in the tumor microenvironment. So the terminology now we use, tumor microenvironment, that's the environment we are talking about. The environment where the cancer cells, a small fraction of tumor cell, collaborate with a large group of stromal cells to orchestrate an environment that helps the cancer cells to grow, right? And we are targeting a small fraction of cancer cells. And while we are dealing with a large population of cells in the environment, we're calling tumor microenvironment, none of them are normal. They're all abnormal in terms of size, in terms of shape, in terms of characteristic of expression of different types of biological molecules, in terms of function. None of them are normal, right? And one of the important component of tumor microenvironment we were discussing in this session is fibroblast, right? And we will discuss how fibroblast will respond to direct interaction from the neighboring cells and also in the context of extracellular particles released by cancer cells and by other stromal cells. So we are focusing on extracellular particles. So before we move on why we should study these particles, we're starting off what are extracellular particles and why it's important in terms of tumorigenesis in the tumor microenvironment, for example, in terms of cell communication. Starting with the basic definition of these particles, is say extracellular particles are membrane-bound particles. So that means the vesicle is composed of lipid bilayers. So that means the vesicle has got a lipid bilayer similar to human cells, lipid bilayer, released by all types of cells. So that means normal cells and cells under stressful condition, whether it's an infected cell, whether they are inflammatory cells, whether they are precancerous cells, whether they are malignant cells, metastatic cells, they all release these particles. And there are different characteristics, there are different types of particles. The most important one, which are studied with big detail, are microvesicles and exosome and apoptotic bodies. There are different types called ectosomes, and I used to work with a specific characteristic of particles. I called them amazome. So it depends on, but focusing on the biogenesis of these vesicles, MVs, standing for microvesicles, with a bigger size, with 100 to 1,000 nanometer in diameter, with an exosome with a 50 to 100 nanometer. And in terms of production method, the mechanism, they have different pathway. Where you can find it in all biological fluid, and that's important. It's very important. Why? We'll discuss it. Why? We can literally now trying to make this story of, so if normal cell release these particles and cancer cell release these particles, at the early stage, at the late stage, at different stage, they're releasing different types of particles. And if these particles can be found in the urine, in the saliva, in the blood circulation, in the lymphatic system, then that helps us to develop a diagnostic procedure, right? This can help us to design a treatment strategy. This particle can be assessed in the urine and saliva to detect the stage of the disease. So the question is how we can characterize these particles, right? So this is the second question. The first question was in the introduction, the second question. So how we can characterize this? Like say, right, this signature is coming from the early stage of cancer, and this is the late stage. Right, guys, one of my student, Amara, who presented the poster presentation, just contacted me asking, is it possible to share the link for the this session? If you can please help with that request. Thank you. Right, in terms of biogenesis, and the composition of extracellular particles. As you can see in this super simple figure, this is a parent cell. Doesn't matter what type of cell. We already mentioned that all types of human cells produce extracellular particles. And the pathway for the generation of vesicles, microvesicles, and exosome are quite different. As you can see, microvesicles are directly budding off from the plasma membrane of parent cell. As you can see here, size are larger than comparing with exosome, but exosome biogenesis is different. In this pathway, the author was trying to make it simple for the understanding, but there are so many questions you can ask. Early endosome, if you define this, how the early endosome is formed, that means when particles are endocytosed, the endosome is formed to contain the materials that is endocytosed within the cell. And after that, after the analysis, they will have two pathways where they're going to be with the multivesicular body formation after the early endosome, whether it's going to be degraded in the pathway if after analysis it will go into the recycling center, or it will be repackaged and sending out. So the question is, if that's the case, then where these contents are coming from? If the cell's receiving some materials from outside and analyzing them, if there are some signaling molecules received for cell proliferation or whatever cell signaling are responsible for, it will be taken, and then the rest will be either going to the recycling center or will be released to the environment. The question is then, in our lab, we found there's so many biological molecules, proteins, enzymes, nucleic acids, lipoproteins can be found in the exosome. So where those contents are coming from. And in terms of the biological function, their function is quite similar to the parent cell. If the cell is a healthy cell, the content of the exosome and microvesicles are biological molecule with physiological function. If this cell is an infected cell, a viral infected cell, the content of these vesicles are particles that is responsible for the infection in the environment, which literally carrying particles if there's a virus or bacteria or other microbial pathogen. If it's cancer cell, then releasing oncogenic factors. So now we can think about this. So the content just literally reflect the physiological function or the pathological function of the parent cell. So this is another question. We can just put it down and say, right, so if a cancer cell try to communicate with cells in the either tumor microenvironment or organs around the body, then they don't need to go themselves. They don't need to break away from the primary tumor and find their way into the circulation and find a secure place and start communication with the environment and ask for a secure place to establish themselves as a secondary tumor. Why? Because if they can manage to transfer and package some oncogenic factors in these vesicles and release it into the extracellular environment, these factors, these oncogenic factors, have the capacity to turn a normal cell into a precancerous cell. So think about this. This could be another method of metastasis, right? With a small size, nanometer in diameter. So we already mentioned the function of extracellular particles. In terms of communication, these particles mediate horizontal transfer of bioactive molecule. Bioactive molecule, we mentioned bioactive molecule, it could be nucleic acid, RNA, DNA, different types of RNA. It could be protein, it could be different types of enzymes, it could be, think about any biological molecule with biological function can be packaged into these vesicles and released into the extracellular environment with a specific postage, we should say spatial delivery, where to go, and which specific cell type they should communicate with. Horizontal transfer. Horizontal transfer means when immune cells, let's say, when immune cells releasing these particles, then if that is a communication take place between same type of immune cells, then that is not horizontal transfer, that's a vertical transfer. But horizontal transfer means immune cell releasing these particles, and then the particles can communicate, establish the communication between the immune cells and the fibroblast, the immune cell and the cancer cell. So horizontal transfer between two specific type from the two tissue type within the body, the communication take place. So these bioactive molecules can influence gene expression. So that means the particles could act as a cargo containing transcription factors, for example. Mediate cell signaling, different types of signaling molecules could be carried out. Signaling molecule, it could be when they influencing immune modulation, pro-inflammatory cytokine, anti-inflammatory cytokines can be released into these particles and then establish communication with the target site, and that influence the immune system function. One of the questions you may ask and say, what is the reason or what's the purpose of packaging this cargo? Like, why a short interfering RNA should be packaged into vesicles and then send it into the extracellular environment? The answer is, this is one of the important questions, guys. You need to think about this. Any type of biological molecule that are not exposed to the immune system before, when the immune system are matured, they will be considered as a non-self, and they will be removed from the circulation, from the extracellular environment, from the lymphatic system. So these particles, most of these particles are the product of the cell that are recognized and considered as a non-self by the immune system. They cannot stay in the blood circulation, in the lymphatic system for a long period of time. If researchers arguing and say, we would like to analyze tumor DNA in the circulation, that logically doesn't make quite sense. Why? Because the tumor DNA is considered as a non-self, as a foreign materials to the immune system. How long should we expect the tumor DNA, which is now we're using that as a marker? When we say marker, that's a unique, specific, characteristic, characterized gene product of the tumor cell. So that means the unique structure. If they have a unique structure, that structure is considered as a non-self by the immune system. We should not expect to observe that in the blood circulation. Why? Because the function of immune system is phagocytose those particles. I'm sure you'll come across with a disease called systemic lupus erythematosus. So how the systemic lupus erythematosus is developed? What's the problem with that? Nuclear antigen. What's a nuclear antigen? Nuclear antigen detected in the blood circulation, and then immune cell recognize that as a non-self, and then inflammation start. So what do you expect in the case of cancer? If cancer releasing those particles, they will be detected. And that detection is not easy, straightforward. One, you cannot detect a high level of those DNA materials in the blood circulation. Second, that leads into the activation of phagocytic cells in the blood circulation, and that causes inflammation, inflammatory response. So we have to think about this. I heard about the research project in one of the universities. They had, I think, a £5 million grant focusing on DNA materials released by cancer cells, use that as a signature marker for the early diagnosis. How you can find it? If the immune system is absolutely, the phagocytic cells are dysfunctional, you can identify those DNAs. Otherwise, it's not possible. Phagocytic cells are there. We will discuss that. So in tumor biology, cancer cell release extracellular particle, more extracellular particles than normal cells. You may think about, well, how many particles can be released by a cancer cell, a single cancer cell. It's estimated that a single cancer cell can release between 1,000 to 10,000 particles per hour. Per hour, right? You just think about this. A single cell, cancer cell, can release, let's say, 5,000. Not 1,000, not 10,000, 5,000 particles per hour. So in 12 hours, where are they going? Once they are released into extracellular environment, lymphatic system, blood circulation, urine, saliva. When we say cancer cell, it doesn't say metastatic cell. when the cancer is at the early stage, they can release this particle, right? And can be identified in the urine. What we are doing now, unfortunately, we are taking blood sample. We're looking for abnormal cells. Abnormal cells. What does that mean? Observing abnormal cells in the blood circulation? Yes. So that means we are not diagnosing early stage of cancer. We design the system to diagnose metastatic cancer. By definition, if cancer is breaking away from the primary tumor and find its way into the circulation, then that's a metastasis, right? That's a metastasis. So if we found a cell or group of cells, abnormal size, shape, whatever, in the blood circulation, in the lymphatic system, then we say, oh yes, cancer. It's not cancer. Excuse me, that's not cancer. It's a metastatic cancer. The system that detecting it is designed to diagnose metastatic cancer. So if we're designing a system to diagnose metastatic cancer, then we should not expect the outcome should be marvelous. Of course, if it's a metastatic cancer, then the mortality rate is, you can talk about that. And when we say 22 million or 35 million people will be diagnosed in 2050 per year, then we're diagnosing metastatic cancer. So what we are doing is just literally causing stress, anxiety to the patient, the relative of the patient, and wasting money to just informing the patient, sorry, you die in a 6 months time, 1 year time, or whatever. So that is the problem, right? This is the problem. Treatment is not the problem. Diagnosis is the problem. So a cancer cell, a single cancer cell can generate up to 10,000 particles per hour. And these particles facilitate the process of cancer genesis, angiogenesis, blood cell formation, invasion of the tumor microenvironment, degradation of the matrix, facilitate metastasis, right? So how these things can happen, I mean, how this can logically make sense that extracellular particles promote tumor growth. What does this mean? It means they're releasing growth factor, and that growth factor binds, once they're released into the extracellular environment, bind by autocrine signaling back to the parent cell, which is a cancer cell, and that enhancing cell proliferation. It doesn't need growth factor from outside, producing their own growth factor. Angiogenesis. Cancer cell in the tumor microenvironment, we said endothelial cell is one of the component of the tumor microenvironment. So cancer cell can produce growth factor, endothelial growth factor, that can activate endothelial cells going on the cell proliferation and differentiation, and leads into the formation of new blood vessel. Invasion and metastasis, cancer cell can enhance the production of degrading enzyme such as matrix metalloproteinase and immunosuppressive proteins such as anti-inflammatory cytokines that can regulate and control the tumor microenvironment for their own advantage, growth, and metastasis. One of the most important aspects of cancer metastasis is epithelial to mesenchymal transition. So in the first slide introduction, we said 80 to 90% of cancer is derived from epithelial tissue. So once the cancer is developed, then if the cancer meant to invade the local stage or the local invasion, or the local metastasis, and then find its way into the blood circulation and lymphatic system, then cancer cell must transform into a specific type of cell which is called mesenchymal cells. So epithelial tissue is defined as a group of cells growing together as a pattern of epithelial sheet. So one of the specific characteristics of epithelial tissue, epithelial cells, are they're growing together. They're growing together. And what's the mechanism of this adhesiveness? Is the high expression of epithelial markers such as E-cadherin. There are many. So this is one of the most important adhesion molecule. They express high level of adhesion molecule in the plasma membrane, and it's acting as a glue, holding the epithelial cells tightly together. So if cancer cell meant to metastasize and breaking away from the primary tumor, then they have to lose itself, break themselves away from the primary tumor site. So how this technically can happen is the downregulation of this adhesion molecule. So downregulation of this adhesion molecule that literally end up the cells to grow independently, which is the characteristic of mesenchymal cell. And extracellular particles released by cells in the tumor microenvironment, not cancer cell themselves. Stromal cells releasing extracellular particles that can help cancer cells to transform from epithelial to mesenchymal cells. And then they downregulate the expression of E-cadherin and upregulate the expression of mesenchymal marker vimentin. So a specific type of biological molecules are identified in the cells from the tumor microenvironment, specifically fibroblast releasing TGF-beta. Different types of ions. We observe calcium ion, high level of calcium ion in the extracellular particles. So these have significant influence on the transformation of epithelial to mesenchymal characteristic. And that makes the cancer cell to be more free, grow independently, motile, invasiveness pattern can be appear when the epithelial cells transform into mesenchymal characteristic. And obviously, that leads into drug resistance as well. You may ask and say how this can happen. When epithelial tissue transform into mesenchymal transition pattern, then how they try to protect themselves against anti-cancer drug. You may see here. So what's happening? We used to work in this research project. As you can see, the top line here are epithelial cells growing in normal medium from day 0 to day 5. And then the bottom part, similar cells, epithelial cells, but co-cultured with cancer microparticles. As you can see, these cells, epithelial cells, under normal condition, growing together, as you can see, patch of cells growing together. Why we already mentioned that, expressing high level of adhesive molecule. And then you see day 5, nearly they're making an epithelial sheet, you see. But when these cells, epithelial cells, are co-cultured with cancer cell microparticles, as you can see, day 3, the proliferation rate decreased significantly. If you start counting the number of cells under normal condition, comparing with the cells co-cultured with extracellular particles, the number significantly reduced. And then you can see some morphological changes. In day 5 is quite evident. As you can see, none of these cells have intention to grow together, comparing to the top picture here. And cells are looking to stretch out. And that's the characteristic of mesenchymal cells. They don't have top-bottom polarity. They don't grow together. And they have front-back polarity, stretched out. And here you can see the staining characteristic. The green fluorescent staining, it's the E-cadherin molecule expression on the plasma membrane of these cells, showing the expression of E-cadherin on epithelial cells, this green fluorescent. And here, nuclear staining, the DAPI staining, and the merging the this E-cadherin staining, the cytoplasm merging with the nuclei. And you can see here clearly showing the expression of E-cadherin on the plasma membrane. While you can observe in this part, the expression of E-cadherin are significantly reduced. Why? Because cells are transformed into a different characteristic. Stretched out, the expression of E-cadherin decreased, and we measured the expression of vimentin, and vimentin expression increased here, comparing with the cells under basal condition. So the question, we said how transformation of epithelial to mesenchymal pattern can protect the cells to resist against cytotoxic drug is showing here. As you can see, the proliferation rate of cells, this is the cell group of cells and the normal condition, you can see the proliferation rate is significantly high comparing cells treated with cancer cells, and they transform cancer cells microparticles, and the proliferation rate remains significantly low. So you can think about if cells meant to not going under proliferation, then they are safer than those cells going under cell division. And then there are many, I'm sure you're familiar with, there are cytotoxic drug targeting DNA replication and then cell cycle arrest and apoptosis. So this is one of the mechanism. Mesenchymal translation can protect cancer cells against cytotoxic effect of the drug. So we move on to, that was about epithelial cells. So now we move on to fibroblast remodeling. Cancer cell microparticles can affect the function and also the morphology of fibroblast as well in the tumor microenvironment. So this is MRC5 fibroblast cells from the lung. In the tumor microenvironment, you just think about if epithelial cells become cancerous. Well, you may ask the question, you say, well, where the pathology start? This is a question we can ask probably later on rather than at this stage. So we assuming pathology start from this picture, we say. Pathology start from cancer cell. Right. Cancer cell communicating the tumor microenvironment has got a certain request. What's the request in the tumor microenvironment? Well, the proliferation rate of cancer is significantly high. So the request is they need space to grow, right? They need space. What else? Nutrient. So two important things, space and nutrient. So communication start with neighboring cells. What are the neighboring cells? Fibroblast is number one. Why it's number one in the tumor microenvironment? The reason it's number one, because this stromal cell facilitate the mechanism of tumor genesis. The mechanism. Why the mechanism? You remember in the first slide introduction, I said, if you analyze a tumor cell, a tumor tissue, 10 to 20% or maximum 30% of tumor tissue is composed of cancer cells. The rest are the stromal cells. And fibroblast is one of the important elements, right? Why it's important? It's important because I don't know how many of you guys working in cells in the lab. My question is, so let's leave this topic here. I take you to the lab. If you grow cancer cell, the most aggressive type of cancer cell you can grow in the lab, in a culture flask. If you leave that for 3-4 days, what will happen? Once they reach the confluency, then they stop growing, right? They stop growing. Change the medium, put fresh medium, exchange, fresh medium, increase the growth factor. Whatever you do, they don't grow. Why? Why they don't grow? What is the reason? What is the specific characteristic of cancer? Uncontrolled cell proliferation. What's happening once they reach the confluency? Uncontrolled. It's become controlled. Stop growing, right? So you can't define that. So what's happening? As a specific terminology called contact inhibition. Contact inhibition. As soon as they become confluent, as soon as the surface of the culture flask is covered by the cancer cells, they're communicating with each other and then sending signals to each other and say, stop growing. Why? Because there's no space to grow. There's no space. Right? So my question is, instead of designing, before proposing my question, please search. You say, we already mentioned that, say, 90% of patients die as a result of cancer is the reason metastasis, as a result of metastasis. Only 10% people die as a primary tumor. And that's like a specific type of cancer. It's a brain tumor, for example. 90% metastasis, right? So here, what we are trying to say is, instead of killing cancer cells, trying to kill cancer cells, why we cannot establish an environment for cancer cells to cannot find a way to invade the neighboring tissue? Why we are not able to put cancer cells in a situation that's sending signals to each other and they say there's no space to grow? How the tumor is formed? My question is how the tumor is formed. I mean, when we say in the lab, it's a super simple question. We say, why did it stop growing in the lab, in the culture flask? We say, well, there's no space. What do you mean space? Surface. Culture flask space doesn't exist. So that means surface is very important factor for cancer cells to grow, right? So if that's the case, then where this surface formation is coming from in the context of tumor? They don't grow on the top of each other. They don't. If you analyze a tumor, they don't grow on the side, on the top of each other. It's just one layer of cells. And then what's happening after that? After that, this cell, fibroblast, laying down ground substance, and then cancer cell coming, growing another layer. And these cells, matrix, producing matrix, cancer cells growing. And that's how tumor is formed, right? So now you can think about this. Fibroblast is, in fact, the major problem. Tumor formation is developed. Yes, we cannot deny cancer cells growing continuously, but there is a limit for them. If they don't have space, they release signaling molecules called contact inhibition, and they stop growing. And you can test that in the lab. And that doesn't happen in the tumor microenvironment. And we keep focusing on cancer cells. Of course we do. But we think about other elements. stromal cells in the cancer microenvironment. So these cells, the fibroblasts, are the stromal cells, and the shape and the size of these cells are specialized, tissue specific, organ specific. They are spindle shape, oval nuclei. The function is production of extracellular matrix. We discussed that. It's not only involved in the tumor genesis during a wound healing, playing very important role. You know, the gap when the physical injury take place in a tissue, when a gap is created and the gap is closed by these cells, what they do, they're releasing collagen. And similar function take place, unfortunately, in the context of cancer. So if you will find this terminology say cancer cell hijack that mechanism of wound healing and the process of the extracellular matrix production for tumor formation. So extracellular matrix production is the key for tumor formation. Modulating inflammation is another important factor in the tumor microenvironment. It could be autocrine or paracrine signalings when we're talking about paracrine signalings, because the signals, the anti-inflammatory signals, are released by, not by this is the primary one. This is the fibroblast and the normal condition. This picture is when the fibroblast is co-cultured with extracellular particles from cancer cells. This is only a morphology analysis. You can see some changes, but the expression of specific markers can be detected here, which is absent in the cells under control condition. And this type of morphology and other characteristic when they're expressing high level of extracellular matrix and other signaling molecule called cancer-associated fibroblast, right? Cancer-associated fibroblast. So that means, so now we are trying to discuss this. The fibroblast you can find in the tumor microenvironment is not the normal fibroblast. It's a cancer-associated fibroblast. And another name, terminology is called myofibroblast, right? Myofibroblast. So these are the important factors that help the cancer cells to form the tumor. So, fibroblast activation and myofibroblast translation it's been accepted widely now. We say if you analyze the tumor and then observe the fibroblast or stromal cells, then you can measure the expression of specific markers that can confirm the fibroblast in the tumor microenvironment is not a normal fibroblast. And it's called cancer-associated fibroblast. What does it do? So here it says cancer cell releasing a specific type of cytokines, interleukin alpha, interleukin 1 beta. There are many signaling molecules that transform fibroblast into cancer-associated fibroblast. once the cancer-associated fibroblast is formed, then what does it do? First function. Cytotoxic T cell, you probably say, oh, what's cytotoxic T cell doing in the tumor microenvironment? Cytotoxic T cell, T lymphocyte. T lymphocyte should be in the blood circulation. Not even in the blood circulation. They're in the lymphatic system, in the lymph nodes sitting down there. Why they are mobilized here? Well, they are invited here. By who? By macrophage. Macrophage. By dendritic cells. Macrophage are residential cells. We're calling community police officer, right? They are there sitting down and looking for some abnormal cells, microbial pathogen, etc., right? So if they observe cancer cells misbehaving, their function is, the function of macrophages, one of the important function of macrophages are phagocytosis of materials that are considered non-self, right? So any type of particles released by cancer cells, if they are recognized as a non-self, they should be phagocytosed. So what will happen after phagocytosis? After phagocytosis, macrophages going up the activation process and then move into the regional lymph node where you can find the cytotoxic T cell. Well, at this stage, it doesn't call cytotoxic T cell. They're T lymphocyte, right? CD4 positive T cell, CD8 positive T cell. Once they inform the antigens, biological molecule released by these cancer cells and identified as a non-self to the body, then will be presented to CD4 and CD8 positive T cell. Then CD8 positive T cell and CD4 positive T cell going under differentiation process. CD8 positive T cell turn into cytotoxic T lymphocyte. Cytotoxic T lymphocyte. They are mobilized from the lymph node into the site. Which site? Where the tumor cells are located, to sort out the problem. What's the function of cytotoxic T lymphocyte? Inducing cell death. Inducing cell. So these cells are mobilized to the site to kill cancer cells, right? They're ready. What's happening here? Cancer cell, before they come, they communicate with stromal cells and make them ready. They don't need to fight. Cancer cells don't need to fight with cytotoxic T cell. Cancer-associated fibroblast is there, right? Break them down. Have you heard about exhausted T cells? If you take a section, tumor section, you can see trillions of T lymphocyte, and none of them are functional. All of them are we call exhausted. How? These cells, cancer-associated fibroblast, make them to keep quiet, and cancer cells keep growing, right? Dendritic cells, another antigen-presenting cell, important antigen-presenting cell. Any type of particles released by cancer cells that mean to be phagocytosed either by dendritic cells or macrophages. Similar function, macrophage, and dendritic cell, they're both responsible for presenting foreign materials. In this case, if cancer releasing oncogenic factors, oncogenic proteins, oncogenic receptors, then these cells and those receptors and those molecules will be identified through this communication of dendritic cells and macrophages with the cells from the adaptive immune system, CD4 and CD8 positive T cell. Why this communication doesn't take place? It does take place in the first step. But when you see a large population of T lymphocyte in the tumor microenvironment, that means they are mobilized from the lymph node into the tumor microenvironment to fight against cancer. They are not there to protect them. So what's happening? Why they are not able to? Have you heard about the techniques nowadays are developed? CAR-T therapy and etc. So they're trying to make these cells ready to go and don't respond even to these cells. And even they don't need macrophages to be communicated with CD8 positive T cell. Right? And these cancer cells downregulate the expression of most of the cytotoxic compound released by CD8 or cytotoxic T cells or other cells coming to the site to fight against cancer. They immediately become neutral. It's not neutral, they're working for cancer. It's like cancer cells. You can see cancer-associated fibroblast. Another important transformation take place. Here we give example. Macrophages, if it's in the early stage, M1 is a specific characteristic that carry out physiological function. Carry out physiological function, that means the function, what's the physiological function of macrophages? The function of macrophages is their community police. If any misbehaving observed, they will be detected. In the tumor microenvironment, they are there. If cancer cell releasing, we give example of between 1 to 10,000 particles are released. In the olden days, scientists believed that they said, no, these particles just are packed with biological waste. But later on, they said, no, they are not biological waste. Macrophages recognized them, said, no, they are not biological waste. They are abnormal. The particles are abnormal. Right? So then what happened? Here you can see transformation take place from M1 to M2. In the tumor microenvironment, this transformation takes place similar to fibroblast and myofibroblast, and similar to dendritic cell and tolerogenic dendritic cells. So this is the problem. You cannot find a single cell, a stromal cell, to function normally in the tumor microenvironment. So why we should focus on cancer cells only? Why we should not focus on these collection, which account for 70%, 80%, depends on the stage of the cancer, of the tumor tissue, and still we're focusing on cancer cells. And unfortunately, we already discussed that when we diagnose cancer, we're looking for cells in the blood circulation, cells in the lymphatic system, analyzing cells with a biopsy and looking how cells look like, how cancer cells look. We're focusing, what about fibroblasts? What about macrophages? What about dendritic cells? What about tumor-associated macrophage and cytotoxic T cell and endothelial cells, parasites, collagen? So these are the important factors need to be considered. If you would like to design drugs, we have to consider the tumor microenvironment. That's the elements. What about the environment itself? Hypoxia, hypoxic condition, acidity of the tumor microenvironment is absolutely disgusted. And one of the, I use the word disgusted because one of the reasons these cancer cells try to move out from the primary site, from the tumor microenvironment, because it's toxic. It's a toxic environment. It's quite normal. I mean, what will happen if a person is suffocated in a room, in a small space? Try to break whatever coming up and try to survive, try to move out. And that's a similar situation in the tumor microenvironment. Extremely toxic, acidic condition, hypoxic condition. So the metastasis is the consequences of the environment that is established either by the cells or probably in some cases we creating an environment, probably coming up here or the next slide we can discuss that. So when we say creating the environment and helping the tumor cells to metastasize, remember this question, please. So the importance of temporal modeling. The interaction of the tumor microenvironment, the instrument cell with the cancer cells, and the matrix components are dynamic. Why? Because it depends on the stage of disease. At the early stage, it's a different communication. When we say it's different, why? Because previous slide we discussed that. Transformation of M1 to M2 doesn't take place at the early stage. Transformation of fibroblast to myofibroblast, it doesn't take place at the early stage. Transformation of dendritic cells to tolerogenic dendritic cell doesn't take place at the early stage. So when these transformation take place, it depends on the stage of the disease, right? So if that's the case, then my question is, if that's the case, why we should focusing only on the characteristic of cancer cells to diagnose the stage of the disease, right? If those components, if 70% or 80% or 60% of the tumor tissue is composed of stromal cells, and those cells are not normal, and transformation of those cells from normal to abnormal doesn't take place overnight, then that means we can identify those cells to stage the disease, if that helps, along with, we're not saying, I'm not arguing here, say, leave cancer cells. No, we should analyze stromal cells along with cancer cells. And that could help us to predict the diagnosis, the treatment strategy, and predict drug resistance and any other phenotypic changes can take place in the tumor microenvironment is a bonus for in terms of designing the treatment and for prognosis or monitoring the treatment. So if you're focusing on fibroblast, the activation of fibroblast in terms of morphology, yes, they become more star-shaped. At the molecular level, they're expressing different type of markers, protein expression, alpha smooth muscle actin expression increased, and fibroblast growth factor expressed higher in myofibroblast, and myocardium related transcription factor A and B, and CCTTA enhancer binding protein beta, and microRNA 21. So these two transcription factor and this microRNA regulation, upregulation can take place and are also observed in the stage of differentiation of fibroblast into myofibroblast. So these transcription factor is responsible, both are responsible for the expression of a skeletal protein that helps the fibroblast to transform into the activated form of the fibroblast, along with the microRNA that binds to the messenger RNA that's supposed to be expressed and maintain the integrity of fibroblast will be downregulated with this microRNA 21. So these are the markers you can use to observe if fibroblast, at least you can see the balance of what the percentage of fibroblast and myofibroblast in the tumor microenvironment is. A 50-50. What does this relate to, the stage of the disease? Of course, we need help from probably AI, something. If there's a 20-80 or if it's 60-40 balance between fibroblast and myofibroblast, what stage of disease could be, how they respond to treatment, what's the function of cytotoxic T cell, if the expression of, for example, myocardium-related transcription factor A and B, and then enhancer binding protein are at this level, or the expression of microRNA 21 related treatment. What stage of disease could be when this expression are at this limit. So the implication of tumor microenvironment, the myofibroblast, the activated form of fibroblast releasing degrading factor, MMP9, matrix metalloproteinase, and pro-tumorigenic cytokines, remodeling extracellular matrix, promote angiogenesis, and facilitate cancer cell invasion. Okay. So fibroblast transform into myofibroblast with the support of, we already discussed that, expression of bioactive molecule from the cancer cells. The fibroblast supposed to protect the tissue. Now they work for the cancer cell. Releasing matrix metalloproteinase. What's matrix metalloproteinase? It's a degrading enzyme. So cancer cell activating fibroblast to degrade create space, degrade the tissue, create space, and that literally helped the cancer cells to grow. So what does this mean? You remember we said if cancer cells, if we can create an environment like a culture flask for cancer cells, they cannot grow. They will stop growing in a stage of releasing contact inhibition. But fibroblast, myofibroblast, releasing degrading enzyme facilitate the process of tumorigenesis. You can observe if the tissue section, cancer cell always located behind the fibroblast. So in some talk, you can probably heard about this. You say cancer cell moving behind the myofibroblast towards the blood circulation. So that means myofibroblast guiding the cancer cells from the primary site towards the lymphatic system and blood circulation. So myofibroblast is the facilitator of metastasis. How? By expression of tumorigenic cytokines and all these degrading enzymes, such as MMP9, matrix metalloproteinases 9, 1, 2, 7, etc. So remodeling the extracellular matrix, how they're remodeling it? Well, once been degraded, removed, they create space, and then cancer cells coming, laying a fresh layer of collagen, and that's a ground substance, scaffolding protein, and that creating space, like you subculturing cancer cells. When cancer cells become confluent in the culture flask, you're subculturing it, and they start growing again. So myofibroblast, that literally subculturing it, creating space, laying down new ground substance, new scaffolding protein, and then they're going under cell division. So cancer-derived extracellular vesicles from A459 cells, that is lung cancer cells, inducing fibroblast activation. So it was done in our lab as well. There's so many papers already published looking at the expression of the alpha smooth muscle actin, fibroblast growth factor, and other markers from the myofibroblast when the normal fibroblast, primary fibroblast is co-cultured with the particles from the cancer cells. As you can see here in this picture, it's showing a normal fibroblast, and these are the factors, biological factors, right? So you can see receptor tyrosine kinase ligand for the fibroblast growth factor, all inflammatory cells here, reactive oxygen species, transforming growth factor, beta, DNA damage, extracellular matrix stiffness, and also this Notch ligand binds to delta receptor. All these biological factors can facilitate, induce, mediate the process of fibroblast activation. Where are they coming from? We give example, we say cancer cells facilitate the process of activation, right, in the tumor microenvironment. But this is very interesting. One of my research project students back in 2020 was working on a research project I'm sure you come across with a situation when they say starvation was a mechanism. I remember there was this approach was used in many countries. Starvation as a part of treatment for cancer, just giving very, very significant low level of food for cancer patient, right? So I was discussing this with one of the a colleague. I asked this question. I said, what do you feel and what do you do when you feel hungry? Right? What do you do? If you're at home, you go into the kitchen, right? If you feel hungry, you're going. If you're in the bedroom, you're going to the kitchen. Right? If you're in the living room, you're going to the kitchen, right? Movement. Feeling hungry, right? Open the fridge, there's no food. What you do now? Superstore. You have to drive. Right? Driving. So if you're starving cancer cells, what do you expect? Do you expect they stay there? No. They're searching for food, right? They're searching for food. Yes, a large population of ordinary cancer cells will die, right? My student was using serum starvation here, the 2.5%. So cancer cells were growing in 2.5%. And here, 1.25%. Then a large population of cells were dead. You can see here the particles and there, but a small fraction, probably 0.001% of that population remain alive. My student put up here, this circle is meant to be this area. So these are the apoptotic bodies. And the question is, what is this, right? And you probably say, no, definitely it's not cancer cell. Or what is this, right? So large number of cells are dead. But what we are currently doing when we are working in the lab, when we analyzing or carry out cytotoxic assay, whatever type of assay we perform, we analyze the effect of cytotoxic compound. So that means we analyze how this compound induce apoptosis or necrosis or whatever. So we analyzing dead cells, right? We analyze dead cells. And we are so excited when we see 90% of cells are dead. And my student was so surprised, excited when we say, 80% of cells are dead when we reduced the serum concentration to 1.25%. And we carried out statistical analysis and turned out significant. So I have a fantastic result. Yes, 80%, 90% cells are dead. It's fantastic. But what about that 10%? So we don't care about the 10%. We managed to kill 90% of cells. So 10%, no problem. In the lab, what we do? We're throwing it in the biological waste. Right? We're analyzing the 90%. We carry out the statistical analysis, p-value, etc., etc. What's happened? Nobody asked about that 10%. Nobody asked about that 5%. Nobody asked about 1%. You say 99% of cells are dead. Fantastic. This is the best drug ever, killing her. What about 9%? So forget about it. No, don't forget about that. Don't forget. Why? Because in the lab, we're throwing that in the biological waste. But in the body, we don't have biological waste. Right? That 1%, will come back. You know, this cell will not die with the concentration of the drug we designed to kill. And these are part of the problem. And we never ever analyze this. And this is what we are doing, literally. Read paper, find a paper, share it with me. Throwing the dead cell and bring 1% of that, or 0.001% of cells, they remain alive, and say how these cells, small fraction of cells, remain alive. Right? This is part of the problem. We need to analyze these cells. They may not be ordinary cancer cells. If we would like to design treatment, we need to design treatment based on these specific cells. They remain alive, not based on those 99%. So this was like a diagram how tumor cell influencing the stromal cell. Once the stromal cells surrendered themselves to cancer cells, then they are under the influence of cancer cells, and then cancer cells don't need to deal with tumor-associated lymphocyte and macrophages type 1 or dendritic cells and other stromal cells in the tumor microenvironment. Once they are turned into tumor-associated, we're calling tumor-associated fibroblasts, tumor-associated macrophages, tumor-associated lymphocyte, and tumor-associated endothelial cell, tumor-associated parasite, then cancer cell is safe. Cancer cell is safe. Drug cannot work. They can resist against drug. They can protect themselves by the help of stromal cells. So we had some FACS analysis, fluorescent activated cell sorting analysis on MMP9. So as you can see, these are the untreated one, and when the cells are transformed to myofibroblast, and you can see literally the expression of the MMP9, and this expression is significantly high, and you can literally compare that with these untreated cells, and even higher here in the EV. That's very, very interesting. The expression of MMP9 is high in extracellular particles comparing with the cell. The cell releasing less EV than the particle containing. So that means extracellular particles considered as an exceptional mechanism of communication that can work with the area, whether it is in the tumor microenvironment or whether it's in the wider circulation. And here we've got another transformation. Imagine if we didn't use this DAPI staining, and if I share this with you and I was saying these are cells treated with extracellular particles from cancer cells, and then it's hard to imagine. I said, I don't think they would be cells. They don't look like cells, but we stain that, as you can see. Although these changes are quite weird. You can see it doesn't look like to be epithelial cell, and this one, and here you can see a naked nucleus, and here, and also this area, all these, you can see transformation is completely changed. So these are the effect of the cancer cell microparticle on target cells. So the actual mechanism, so you probably also say how these communication take place. So how these vesicles are absorbed by the target cells. We don't have time to discuss that. Hopefully we will discuss it in the future. So this is a current project, research project. My MSc student, Amara, she presented her work yesterday, I think, poster in the conference. She's working on this project. And as you can see, the extracellular particles carrying different concentration of calcium and magnesium, and also we observed a very, very specific characteristic changes. As you can see, they release different population. So here, this is the control. This is the population of extracellular vesicles here. And these two are different condition. So we are focusing on this population, right? So this population is a very low level of site scatter here. And again, here, the population changed. But what we are planning to work and identify, if these vesicles are released by cancer cells, then how can we design a platform to identify these particles, let's say, in terms of, I'm not very familiar with AI platform to identify this population in the blood circulation, in the lymphatic system. So we have to, Amara's working on the culture fluid. So it's a vitro work. But we need to observe this population in patient sample, in the serum sample of patient. And then if we can identify this population of vesicles, and we can characterizing it at the early stage, let's say, when we say stage 1 and stage 2, stage 3, stage 4 of cancer, or we discussing this, whether it could be when the treatment start, how they're releasing this, what's the content of these vesicles, and this in terms of size, shape, characteristic, you can already see it's different, and this is quite exciting. If you ask question, I don't know the answer, because we found this. It's a change. We have to work on this, and we need to use transmission electron microscopy analysis, content analysis, and we need to use probably AI. I'm not familiar with the technology to create a platform, and then from there you can literally design a kit that can be test like a similar to pregnancy test, similar to taking a blood sample and or urine sample and or saliva, and you can detect these vesicles. And if that's the case, then we can literally identify a tumor that is developed and reside in the primary site far, far early than cancer cells disseminated from the primary tumor into the blood circulation, which is literally useless. Apart from wasting time, money, creating anxiety for the patient, and start aggressive treatment, nothing is achievable, right? So what else we did also here in the gel electrophoresis, we found the band. This is the EV analysis, and that was quite strong. And this one, it's thin, but this is quite thick. And these are the parent cell, and this is the extracellular particles, which is literally, so these are the ladder. We found this. We could analyze, put this along with the EV's analysis to finalize what is this specific signature. And I personally think analyzing extracellular particles would be the ultimate answer for early diagnosis and a better treatment and a better patient outcome. And here, as you can see, might not be very clear. You can observe cells are going under apoptosis. As you can see, blips are everywhere, here and there. So these two slides are from cells they are under stressful condition. They couldn't manage to survive, and they are literally are probably at the early or late stage of apoptosis, as you can see. But in the meantime, we've got individual cells in the same treatment part, which are still have no effect. As you can see, the cell membrane is intact, but the morphology is completely changed. So these are the cells needs to be analyzed and needs to be going through the gene sequencing analysis and observe, isolate extracellular particles from these cells, how they react. If they mean to inducing resistance, these are the cells need to be investigated. So I think this is the yes, the last slide. And thank you very much. I really appreciate. I think it was a bit too long, and I am here to answer any question, guys. If you would like to ask, if I use the, let me check the chat box.
Thank you, sir. Your session is such an amazing and insightful topic. So with your concern, I would like to present some questions from our participant.
Sure.
So our first question is, are there specific signaling pathways identified that mediate the fibroblast extracellular particle crosstalk?
I didn't share some of the work we had in the previously already discussed that. So the signaling pathway the first question I should start with is, what's the mechanism if the particles bind to the fibroblast, then the question is how these vesicles are endocytosed. So this is the question. Probably we say, what signaling pathway involved in this? I would say, well, the interaction take place through the communication with extracellular particles, right? So if the extracellular particles mediating these changes, then how the extracellular particles communicated. So I'm trying to take the guy who asked the question back to say, which element of the particles causing the changes? Is this microvesicles or is this exosome, right? So signaling pathway, just leave that on the side. So the mechanism of communication is the particle is endocytosed by the recipient cells, or it's not endocytosed, it's the fusion of the particles with the recipient cells. My initial response is, I didn't, so I measured before coming back to the signaling pathway. We observed fusion. We observed recipient cells. In day 5, we see a patch of microparticles on the fibroblast. After 5 days, because we stained the microparticles, after day 3, 4, and 5, we saw the stained microparticles become part of the plasma membrane of the target cells. So we found fibroblast growth factor was involved in the process of the activation. So the signaling pathway was activated through the expression of fibroblast. That was growth factor released by the cancer cell.
Sir, one more question is there. What roles do fibroblasts play in tumor progression and microenvironment modeling?
We already discussed that in the discussion. The picture, when we put through and we discussed that, fibroblast, as we mentioned, that is the one of the most important cell in the in the matrix, in the stromal cells in the tumor microenvironment. So what role it plays? As I mentioned, fibroblast, I give example of if you work in the lab and grow cancer cells in a culture flask, what will happen? So when cancer cells grow and then culture flask and the cells are enriched in the confluency, there is no space for the cancer cells to grow, then what will happen? Cancer cells stop growing. Then researchers try to subculture the cancer cells into a new culture flask, create more space for the cancer cells to grow. So the function of fibroblast, I use this terminology, I said subculturing function. So what does literally do? Releasing extracellular matrix, degrading enzymes, and anti-inflammatory cytokines in the tumor microenvironment. When the immune cells trying to come into the area to fight against, for example, cancer, we'll give example of cytotoxic T cell, for example. Cytotoxic T cell mobilized from the lymph node to the site where the tumor is residing to fight against cancer cells. So what happened? As soon as they reach to the site, what does fibroblast do? We should not using the word activated fibroblast or tumor-associated fibroblast. So tumor-associated fibroblast literally using the PD-L1 and PD-L, so programmed dead ligand receptor communicating with the ligand and turning off the function and suppressing the function of the cytotoxic T cell to act against cancer cells. So downregulate the function of cytotoxic T cell, protecting cancer cell. In the meantime, creating space by releasing degrading enzyme. We mentioned that in the last slide, MMP9 expression. That's a degrading enzyme, creating space for the cancer cells to grow. So create a safe zone for cancer cells to grow. This is the main function of fibroblast, or I should say myofibroblast, in the tumor microenvironment.
Thank you, sir. So thank you for such an amazing session, and your knowledge shedded the light on the complex interaction within the tumor microenvironment, which may help us to understand the cancer progression in a very deeper way. So thank you so much, sir.
Thank you. Thank you. Thank you for the team to provide this opportunity. I'm looking forward for this conference to engage more and bring our more students and researchers to talk and collaborate science.
Definitely, Dr. Ahmed. It was nice presentation. I think we need to plan a meeting to discuss further some of the things. I will not talk here at the moment, but we will have a meeting on that. And definitely there are some things which need to be taken further. So we will have that meeting.
Brilliant. Thank you very much, Dr.
Thanks
Praveen.
for the.
I really appreciate for giving this opportunity. We communicate with scientists. Brilliant. Thank you very much. Have a good, I don't know, day here. Evening here. Sorry, it took a bit long, I think. Didn't go according to plan.
Yeah, it's fine. It was a little bit longer, but I think it was needed because it was connecting the all the slides. So definitely it was a good session. Thanks.
Thank you very much. Thank you. Thank you.
Okay, so now I feel very privileged to welcome Dr. Praveen Bade sir, who will be now announcing the prizes and delivering the vote of thanks. Dr. Praveen Bade sir's presence adds a great value to this session. So we are very grateful for him to have, we are very grateful to have him with us. Now, with a great respect, I would like to welcome Dr. Praveen Bade sir to kindly take over the stage.
Thank you, Safa. So we are at the end of the 2 days annual Swalife conference, where we had sessions and poster presentations. So to announce the posters, so totally we had 18 posters. But unfortunately, one of the poster presenter was not able to do a poster presentation. So total 17 posters were present. Out of that, actually, initially we decided that we will put everyone in the same category. But now we decided that we will be actually having 2 different categories. One will be for student and one will be for the faculty. So in the student category, one poster which actually have shown novel chemical synthesis, docking validation, and in vitro activity was the one which actually has created impact. And also, the theme of this conference was about innovation in drug discovery. So definitely that work and the presentation was also done very nicely. So the winner in the student category for best poster award is Kalyani Thombre from Kishoritai Boyer College of Pharmacy, Kamti, Nagpur, for her work on innovative 7O substitute Christine derivatives, where she actually looked for VGFR2 inhibitors. I think the work is promising, and definitely, when we discuss um, about the work, it was having originality. It was also having a significant translational into the innovation and strong scientific execution. So in the student category, Kalyani Thombre poster has won. In the faculty category also, we have 5 posters. In that, one of the poster presentation which we found was very scientifically, it was very creative, had practical applications in drug delivery, and also it was having a clinical relevance. So the award in the faculty category goes to the formulation and evolution of Polyherbal ethosomes gel. So that's Dr. Shashikala Metri from Gopuraju Rangaraju College of Pharmacy. Thank you, Madam, for your presentation and submitting the poster. So these are the winners for our 2 days conference. Over to you, Safa.
Okay, thank you, sir. Thank you, everyone, for attending the conference.
Hello. So just to give a last vote of thanks of this conference on behalf of Swalife Foundation, then Swalife Society for Translational Science and Innovation, and on behalf of Swalife Biotech Limited, I like to thanks for attending this 3rd annual Swalife International Conference on Innovation in Drug Discovery. First and foremost, I like to extend deepest gratitude to our all keynote speakers. Yesterday's Dr. Enrico, the founder of OACP, he talked about genetic biomarker and how to use fish in accelerating drug discovery innovation. We have collaboration with Enrico, and we are actually working on early diagnosing the cancer. Simultaneously, today's keynote speaker, Dr. Ahmed, he is one of the senior lecturer from University of Staffordshire. We actually have the connection when he was doing his PhD and I was doing masters. I know him from that time. His talk on the temporal modeling of fibroblast response to cancer-derived extracellular particles was the very fascinating, and definitely early diagnosing is very important. So we truly honored to have benefited from their knowledge and vision. I heartfelt thanks to all the poster presenters, students, researchers, and the faculty for showcasing their innovative work. Your contribution reflect the vibrant spirit of the scientific discovery and also the collaborative growth this conference stands for. Special congrats to the winners. I would also like to sincerely thank our organizing partners for their constant support. Our gratitude also goes to the organizing committee, volunteers, and the technical team who actually help, and the efforts were very, uh, very important to smoothly execute these sessions. So finally, a big thank you all our participants and attendees who attended from different part of India and also out of India. Also the active engagement, the questions which were put in front after the sessions were really insightful. So it was very interactive and impactful. With this, I like to conclude the proceeding and definitely looking forward to collaborate in future and also to work together on different groundbreaking innovations in drug discovery. Thank you all and have a wonderful evening ahead. Thank you all.